So first and foremost, welcome everyone to the fifth annual and very first virtual Mid-Atlantic Biomaterials Day. My name is Michael Len, and I'll be your MC for the day as the outgoing president of the Johns Hopkins University Society for Biomaterials chapter. On behalf of our entire chapter, I would like to officially extend our thanks to all of you for attending this event. The field of biomaterials has been a developing discipline for many years now, and it really is extremely exciting to see and hear how far the field has come. This growth really reflects on the original goal that we had when we were developing our SFB chapter and our hosting of MABD in general. We started with the idea to help expose students to the field of biomaterials and understand the careers that are available to them and let them learn and know how biomaterials can affect their future lives. As one of the only SFB chapters that's composed primarily of undergraduate students, we've always wanted to place an emphasis on the foundational learning aspect of biomaterials, and we always hope to enrich our efforts with the annual Mid-Atlantic Biomaterials Day. Although our chapter has had its ups and downs in terms of both the membership and activity, I'm honestly proud to say that this year especially, I've had the pleasure to work with one of the best executive boards ever. And this event definitely would not have been possible without all their dedication. Now, through MABD, we hope to extend our reach and influence to help students in the field of biomaterials. And although the pandemic has definitely caused some unexpected consequences and troubles throughout the past year, it has definitely allowed us to open up this conference for the first time ever to a national audience, since we've actually been able to reach out to SFB chapters across the country. And our attendees this year will feature prominent colleagues and guests from the Arizona State University, the University of Washington, and Iowa State University as well. Hopefully, the extensive reach of MAD can continue into the future through even greater collaborations in person. This year's MABD theme is Unveiling Biomaterials Through the Eyes of Apprentice to Master. And although this seems a bit cryptic, our goal this year was really to provide a conference that's beneficial to students on all levels, from the undergraduate who's really just getting to know what a biomaterial is, to the professors that have dedicated years of their lives to crafting and perfecting their art. Integral to this, of course, are our esteemed speakers, including our keynote speaker, Dr. Scott Wilson from JHU, as well as all of our plenary speakers, Ilon Heylesius from Stryker, Dr. Raleigh Linville from the Searson Lab, AJ Bezub from the Paul Corporation, Dr. Christopher Sowie from the FDA, and Dr. Joshua Doloff from JHU. In addition to these outstanding individuals, we will also be featuring undergraduate and graduate rapid-fire research presentations and poster sessions, where the students will be able to showcase their work and demonstrate their talents, with a special prize being awarded to the best in show and runner-up presentations. This year, MABD is proudly sponsored by the Alumni Association and the Department of Material Science and Engineering. Again, thank you all for coming to this event, and I hope it'll be a productive and fruitful experience. So our first keynote speaker for this year is Dr. Scott Wilson. Dr. Wilson recently joined the Biomedical Engineering Department at Johns Hopkins. Earning his PhD at Georgia Tech before embarking on his academic journey, Dr. Wilson has worked with Dr. Jeffrey Hubel as a postdoc and has developed a biomaterials-based therapy for the treatment of celiac disease that is currently in a phase 1 clinical trial. Additionally, Dr. Wilson has recently received one of the Pharmaceutical Research and Manufacturers of America Foundation's 2021 Research Starter Grant in drug delivery for his inspiring work in groundbreaking research. An exciting new professor at Johns Hopkins, ladies and gentlemen, it is my pleasure to introduce our keynote speaker, Dr. Scott Wilson.
Thank you, Michael. Thank you so much for your introduction, and I would like to thank the board for inviting me to speak today. Really impressive undertaking, and I've been very impressed with the organization and the website you all have built for this conference. Can you see my slides, Michael?
I think we saw them for a brief second, but
Okay,
I think they're not there anymore. Yeah.
Okay. How about now?
Yep, that looks good.
Great. All right. So can you see the presenter view or the?
Yeah, we're looking at the presenter view right now.
Let me switch. Here we go. All right, there we go. So today I'll be speaking about some work that we've been doing on strategies for manipulating the adaptive immune response to treat and prevent diseases. And since we'll be discussing the adaptive immune response, and we do have, a very diverse crowd here today for the presentation, I'm going to give a brief primer on adaptive immunity. And adaptive immunity is the mechanisms, a collection of mechanisms, by which the body provides protection in the form of immunity against things like viruses, bacteria, parasites, and cancers. In addition to these quite beneficial roles of the adaptive immune response, the adaptive immune response can also mount an immune response to the patient's own host tissues, causing tissue destruction and a diverse set of pathologies commonly known as autoimmunity. Now, in the case of the induction of immunity for protection, the adaptive immune response is initiated primarily in sites of the secondary lymphoid organs, such as lymph nodes and the spleen. And here in the case of immunity against something like a viral pathogen, the adaptive immune response is initiated when something like a virus, this pathogen, is taken up by an antigen presenting cell. Now, upon recognition by this antigen presenting cell, these viruses are endocytosed, and then they're broken down into their fundamental components, including the proteins that compose the virus. And those viruses and those viral proteins are then degraded into peptide epitopes. And these peptide epitopes are presented on the surface of these antigen presenting cells and can be recognized by viral specific T cells. Now, in addition to recognizing this first signal, which is this viral epitope, these T cells can be activated upon recognition of secondary signals or pro-inflammatory signals, which are produced by these antigen presenting cells. Now, upon recognition of these two signals, these viral specific T cells then become activated, and they can differentiate into various effector phenotypes. Now, in the case of CD4 T cells, these cells can differentiate into helper T cells. And as the name implies, some subsets of these helper T cells can produce signals which activate B cells, which are the antibody producing cells in the body. Then these B cells produce antibodies that disseminate through the host, attacking the viral pathogen, attaching to it, and causing its elimination from the body through a process that I think we are all well aware of now, commonly termed humoral immunity. Now, in the case of Cd8 T-cell activation, once they recognize their viral epitope on the surface of antigen presenting cells and these pro-inflammatory signals, they can differentiate into cytotoxic T cells. These cytotoxic T cells produce cytokines and other cytolytic molecules that are able to destroy virus-infected cells, removing another source of virus within the body. And this process is commonly referred to as cellular immunity. Now, in the case of the induction of autoimmunity, the organs within our body are constantly releasing proteins, commonly referred to as autoantigens, and that can also be taken up by antigen-presenting cells. And then due to various environmental and genetic factors, these autoantigens can be presented to self-specific T cells in concert with other stimulatory signals, causing these self-specific T cells to activate and mount an immune response against the patient's own tissue, resulting in tissue destruction, loss of function, and autoimmunity. So these are the two areas that my lab is working in. So we're working on strategies to turn immunity off and on. And the space that we want to play in, or that we do play in, are these interactions between antigen presenting cells and T cells, so that we can guide these T cells towards a phenotype that treat and prevent diseases. Now, half of the lab, my laboratory, is dedicated to strategies for turning immunity on, where we're taking pathogen and tumor-derived proteins and some biomaterial-based delivery strategy to target these proteins to antigen-presenting cells, along with other immune stimulatory signals, so that these antigen-presenting cells produce the signals that are necessary to activate these T cells, driving them to mount an immune response against things like viruses and parasites, as well as cancer tumors. Now, my lab, the other half of my lab, is really dedicated towards strategies for turning immunity off. Where here we're going to take autoantigen proteins or allergens, plus some immune suppressive biomaterial-based platform to target antigen-presenting cells, so that these cells produce immune suppressive signals that actually turn off the T cell responses that cause autoimmunity. Thus, not just treating the symptoms of autoimmunity, but actually attacking these diseases at their source, at these T cells that drive the deleterious immune responses. So what I'm going to focus on today is talking about a strategy that we've developed using a glycoadjuvant, synthetic glycopolymer, for turning immunity on, specifically for infectious diseases and cancers. Now, our goal was to produce a new type of protein subunit vaccine. So protein subunit vaccines are great in that they're safe and they're easy to produce and they have a long shelf life. And if we're thinking about the current vaccine strategies that have been rolled out for SARS-CoV-2, something that's very quickly produced and is easy to store and ship around the nation, we can see that these platforms do have a lot of advantages that have a real world impact. And perhaps everyone on the call today has received some sort of subunit vaccine platform, whether it be for something like hepatitis or whooping cough. And these are really two component vaccines. So they have two fundamental components. The first is an antigen or protein that's taken from the pathogen to which you want to induce immunity. So a subunit protein. And when those are injected into the host, they're taken up by antigen presenting cells. Here I'm showing a dendritic cell. And these dendritic cells take up these antigens and then they process these antigens and present the peptides from those antigens to both Cd8 and Cd4 T cells. Now, when these Cd8 and Cd4 T cells recognize their cognate antigen, it causes these T cells to proliferate rapidly to undergo clonal expansion, producing an army of T cells that are able to fight the infection. However, this alone is not enough to induce an immune response. So for this reason, subunit vaccines have a second component known as an adjuvant. And these adjuvants, sometimes called danger signals, activate these antigen presenting cells. So these antigen presenting cells then produce the co-stimulatory signals that are necessary to activate these expanding T cells. So in the case of CD4 activation, you get the induction of helper T cells that induce signals that activate B cells through to generate humoral immunity. And current subunit vaccine platforms are actually really good at the induction of humoral immunity. However, they're much less effective at the induction of CD8 mediated T cell immunity. And what this has been attributed to is the inability of current FDA-approved adjuvants to provide the proper dendritic cell activation so that these dendritic cells produce the signals that are necessary to activate Cd8 T cells. And then the second reason is that when soluble antigens are taken up, dendritic cells preferentially present those antigens to CD4, not Cd8 T cells. So in order to build a better protein subunit vaccine, we need to come up with a strategy that we can enhance antigen presentation to Cd8 T cells, as well as increase the activation of these dendritic cells so they produce the proper co-stimulation to activate CD4 and Cd8 T cells. So given that we have a problem statement now, we come up with a set of design criteria so we can build a biomaterial that is actually able to accomplish this goal. So what we recognized from the literature is that because viruses and other pathogens are decorated with glycosylations such as mannose, our bodies have evolved with, the dendritic cells have evolved with mannose receptors that specifically recognize mannose on the surface of pathogens. And then when they take these pathogens up or endocytose these pathogens via the mannose receptor, these antigens from this pathogen are preferentially trafficked through intracellular pathways that preference CD8 antigen presentation. So our idea was, well, let's deliver our vaccine via the mannose receptor. Next, we wanted to focus on activation of these dendritic cells. And so since our vaccine platform is going to be endocytosed via the mannose receptor, we looked at an endosomal activation pathway for activating these dendritic cells. And what we focused on was a molecule called toll-like receptor 7 or TLR7. And activation of TLR7 causes dendritic cells to activate and causes them to produce signals that activate T cells. So we decided to focus on delivering a molecule that activates dendritic cells via toll-like receptor 7. Next, we wanted to deliver an unmodified antigen. So we have these protein antigens, and we didn't want any chemical modification to this antigen because these antigens are taken up by dendritic cells, and then they're processed into smaller peptide units. And if there's chemical modifications on these peptides, it could block loading into the machinery for antigen presentation, as well as block the ability of T cells to recognize their antigen. And then finally, we wanted it to be very small because these things are injected subcutaneously or intramuscularly, and then vaccines need to drain to the secondary lymphoid organs, such as lymph nodes. And so a much smaller construct will facilitate this drainage into these lymph nodes, the source or the location of T-cell education. So with that in mind, we just drew a little cartoon thinking about, well, how could we accomplish all these goals and fit all these criteria into our materials? So the idea that we came up with is that we'd take protein antigens and we would chemically conjugate them to a linear water-soluble polymer. And these water-soluble polymers would be decorated with mannose residues, as well as TLR7 activating molecules. And that we'd attach this polymer to our antigen via self-immolative linkage that degrades once it's inside of the cell. So the idea here is that our polymer would be able to target mannose receptors on the surface of dendritic cells. And then after taken up into the endosome, we would be able to activate TLR7 via the TLR7 ligands that were engineered into the backbone of our polymer. And then within the endosomal space, our self-immolative linkage would degrade, releasing our antigen in its free form. So what we would then get is unencumbered antigen presentation to both CD8 and CD4 T cells, as well as the induction of these co-stimulatory signals derived from dendritic cells that are necessary for activating T cells. So now that we have kind of this idea, all we need to do is come up with this linear polymer and a way to conjugate it to antigens. So in order to generate these water-soluble polymers that have this multiple functionality, we used a Raft polymerization technique, and that's a free radical polymerization technique that's very nice because you can incorporate molecules with a lot of different functional groups into a polymer. So our polymer was made up of these three monomers. So we had a biologically inert monomer, and then we had a monomer that was decorated with this mannose residue for targeting dendritic cells. And then we had a monomer that was decorated with this TLR7 ligand. What you can appreciate is this end of our monomer unit is very similar to other TLR7 and TLR8 molecules that have been previously published. And these molecules are very immune stimulatory. However, they're quite systemically toxic and they have poor water solubility. So by targeting them to dendritic cells, we can actually reduce the toxicity of these types of immune stimulatory agents, as well as incorporating them in a water-soluble polymer. We're increasing their solubility, making them easier to deliver and work with. So the fourth component of our polymer is this Raft agent. And so our polymer is actually going to grow from this molecule, and this molecule then becomes ingrained into the backbone of the polymer. And so that each end of this molecule will be at either end of our polymer. So what we did is we decorated this molecule with this azide functionality that will allow us to attach it to a protein at a later date. So we simply form the free radical polymerization, and we get this random copolymer that has a TLR7 ligand, as well as mannose incorporated into the polymer structure. So as I mentioned, antigens are proteins derived from pathogens. So as a protein, it has at least one terminal amine and several other amines if it has lysines in the structure of the protein. So what we came up with is, in order to attach our polymer to antigens, we came up with this bifunctional linker. And so this bifunctional linker has an azide reactive moiety at one end, and then an amine reactive group at the other end. So we can simply carry out this reaction at room temperature at fairly at a pH of 7.8, so near physiological pHs. And upon conjugating this linker to our antigen, we design this linker so that we form this self-immolative linkage in the backbone. So what happens is inside of a cell, this disulfide bond gets cleaved, releasing a free thiol that undergoes an intermolecular interaction, actually cleaving this linker from the antigen and releasing our antigen in its unmodified form. So we take our linker antigen conjugates and we simply mix them with our PMAN TLR7 polymer, and we can form this full conjugate system where we have our PMAN TLR7 linked via self-immolative linkage to our antigen. So next we asked, are we actually able to deliver our antigen in its unmodified form within a cell? So what we did is we formed conjugates with our PMAN TLR7 system conjugated to the model antigen ova albumin. And then we treated bone marrow derived dendritic cells with these conjugates. And then we lysed these bone marrow derived dendritic cells, and using Western blot, we were able to monitor the molecular weight of these conjugates over time. So if the polymers are in fact being cleaved from our ova albumin, we should see a decrease in the molecular weight of our conjugates over time. So here are the results of those studies. You can see here's the initial molecular weight of our polymer. We can see that after 30 minutes, we do start to see some decrease in the molecular weight of our conjugates. And if we follow this over 180 minutes, after 180 minutes, you can see that the molecular weight of our conjugates is identical to the initial molecular weight of unmodified ova albumin, indicating that we are actually able to cleave, these polymers are actually cleaved in the intracellular environment in physiological conditions. So the next question we asked is, are we able to target dendritic cells in vivo, since those are the cell type we want to target these constructs to? And are these constructs actually collecting within the lymph nodes, which is the site of T-cell education? So what we did is we formed conjugates with fluorescently labeled ova albumin, and then we conjugated that to our PMAN TLR7 polymers. And then we inoculated animals in the draining foot pads with these fluorescently labeled conjugates. And then we isolated the draining lymph nodes from these animals. And then using whole organ fluorescent imaging, we looked at the fluorescent signal in each one of these draining lymph nodes. Now, here are the results for those studies. And you can see that the red bars represents animals that were treated with fluorescently labeled ova conjugated to PMAN TLR7. And as our control formulation, we had fluorescently labeled ova not conjugated to the PMAN TLR7. And as you can see, for the draining lymph nodes, we see a significant increase in the fluorescent signal in animals that were treated with our full conjugate system, indicating that our polymer system is in fact able to enhance the delivery of ova albumin or our antigen to these draining lymph nodes. Next, to look at the dendritic cell subsets that might be targeted by our polymers, we took these draining lymph nodes, and then we isolated the cells from these draining lymph nodes using mechanical disruption. So we're able to get a single cell suspension of the contents of our draining lymph nodes. And then using fluorescently labeled antibodies, we're actually able to label these different cell types within this lymph node single cell suspension. So here what I'm showing is labeling with CD11C, which is a marker that's specific for dendritic cells. So using these fluorescently labeled signals and flow cytometry, we're actually able to measure the dendritic cells within these draining lymph nodes, and more specifically, the activation state of those dendritic cells, as well as determine whether or not these dendritic cells had taken up our fluorescent conjugates. So here are results for the percentage of ova-positive dendritic cells for each one of these dendritic cell subtypes. And all I'm going to say about these dendritic cell subtypes is some of them preferentially present to Cd8 T cells and others to Cd4 T cells. But what you can appreciate in all of these DC subsets, by the increased percentage of ova-positive cells with the red bars as opposed to the blue bars, we can see that conjugating PMAN TLR7 to our fluorescently labeled ova causes a significant increase in the percentage of ova-positive dendritic cells for each one of these subsets. As I mentioned before, we can also look at the activation state of these dendritic cells by looking at their expression of co-stimulatory molecules such as CD80. And here our full conjugate system is shown in the dark gray bars. And here again, you can see that the dendritic cells taken from animals treated with our full conjugate system show increased expression of this co-stimulatory signal, indicating a higher level of activation of these dendritic cells. So we have our two signals that are necessary for T-cell activation. We have antigen being delivered to these dendritic cells, as well as the increased expression of co-stimulatory molecules. So after these studies, we decided to perform an in vivo vaccination study where we wanted to determine whether or not all these cool functionalities that we had engineered into our polymer system were actually improving the efficacy of our vaccine. So what we did is we compared the immunogenicity of ova albumin conjugated to our PMAN TLR7, our full polymer conjugate system, to different constituent vaccine formulation that lacked a single functionality of ova PMAN TLR7. So one of the groups that we analyzed was ova not conjugated to PMAN TLR7 to monitor the effects of conjugating our polymer to the ova. A third group was ova conjugated to a polymer that just contained mannose So we removed the TLR7 from the polymer, and then we administered that TLR7 as a soluble formulation admixed with this OVA PMAN polymer. Next, we had OVA plus just the PMAN TLR7 molecule that we were using in our polymer. And then finally, a conjugate with OVA albumin conjugated to a polymer that lacked mannose, so lacked the ability to target dendritic cells. We vaccinated animals on day 0 and day 28. And then on day 35, we sacked these animals and then looked at the OVA-specific immune response in the draining lymph nodes and spleens of these animals. So first of all, looking at our Cd8 T-cell results. So this first graph here shows the percentage of OVA-specific Cd8 T-cells within the draining lymph nodes of these animals at day 35. And the first column here represents animals that were treated with our full conjugate system, so the OVA conjugated to PMAN TLR7. And then the rest of the groups are the constituent formulations that I showed on the previous page. And what you can see here is our full conjugate system is outperforming the constituent formulations in terms of the induction of OVA-specific CD8 positive T cells. We also looked at cytokine production by these cells because that's an indication of the activation state of these CD8 T cells. And then we can see in the draining lymph nodes, we see increased percentage of cytokine producing cells in the lymph nodes of animals that were treated with our full conjugate system as compared to the constituent formulations. We also looked at the CD4 T cell population from our draining lymph nodes. And when we re-stimulate these cells ex vivo with antigen, what you can see is that we see an increased percentage of interferon gamma and TNF alpha producing cells in animals that were treated with our full conjugate system as compared to the constituent formulation. So these results indicate or demonstrate that our full conjugate system is outperforming the constituent formulations in terms of CD8 and CD4 T cell activation. So moving forward, we focused on the humoral immune response. And what we looked at here is we looked at a subset of T cells which specifically provide assistance to the activation of B cells and maturing B cell responses. So what you can see here, these are known as T follicular helper cells. And in animals treated with our full conjugate system, looking at the spleen, we see an increased percentage of T follicular helper cells in those animals as compared to the other animals receiving vaccinations. And then we looked at the actual concentration of anti-OVA IgG within the serum of these animals. So these are OVA-specific antibodies that have been produced in the blood of these animals. So we isolate the serum, and when we perform an ELISA to look at the concentration of antibodies, we see an increased antibody titer in animals that were treated in our full conjugate system as compared to the others. So based on these results, we see that we're seeing a more robust CD8, CD4 T cell response, as well as humoral immune response in animals that were treated with our full conjugate system. So OVA albumin is a nice test, and we have a lot of molecular tools for looking at immune responses to OVA. However, that's not a model for any real world disease. So we decided to focus on strategies for inducing immunity to diseases that remain a major challenge in the world. And of course, malaria remains a major global health problem, specifically in regions near the equator within the globe, sub-Saharan Africa, South America, and Southeast Asia. So there currently is a vaccine for malaria. It's known as the Mosquirix vaccine. And this vaccine induces an immune response to this circumsporocyte protein, or from now on, I'll refer to it as CSP. And this is a protein that's found on the bugs that cause malaria. And the Mosquirix vaccine actually just uses peptide epitopes from this circumsporocyte protein. So it's not the full-length circumsporocyte protein. And so they've just isolated immunodominant epitopes from that protein. So that's the antigen. And for an adjuvant, they use this AS01E formulation, which these are liposomes that are formulated from MPLA and QS21. And those are just immune stimulatory molecules that activate toll-like receptors. And this vaccine platform, although it was held as a major advancement, and it certainly is, it's only efficacious in about 30 to 50% of patients, with children being the least protected. and it induces nominal CD8 T-cell responses. So we wanted to do a vaccine study to compare our glycoadjuvant to the adjuvant that's used in the current Mosquirix vaccine. So what we did is we vaccinated animals on day 0 and day 21. And these animals either received CSP, the circumsporocyte protein conjugated to pMan-TLR7, or CSP admixed with the liposomal formulation that was like the liposomal formulation used in the Mosquirix vaccine. We also had CSP mixed with a commercially available TLR7, TLR8 ligand, as well as CSP mixed with poly-IC, which is a toll receptor ligand that's been shown to induce immune responses when admixed with CSP. So looking at the results of these studies and looking specifically in the lymph node and looking at the CSP specific CD8 T-cell response, we're here on the y-axis, we see the relative number of cytokine producing cells in the draining lymph nodes of these animals upon re-stimulation with the circumsporocyte protein. You can see that only our full CSP pMan-TLR7 conjugates were able to induce a significant CD8 T-cell response. When we looked at the CD4 T-cell responses, again, looking at the relative number of interferon gamma or cytokine producing cells in the draining lymph nodes of these animals, you can see while the current state-of-the-art CSP plus ASO1E formulation does an admirable job of inducing a CD4 T-cell response, our vaccine platform produces a more robust T-cell response than the current state-of-the-art. So moving forward to the humoral immune response, looking in the blood of these animals, and here in the serum, looking at the CSP-specific antibody response, you can see that the CSP pMan-TLR7 produces a more robust antibody response within the serum of these animals as compared to CSP mixed with the current state-of-the-art adjuvant formulation. So we have a more robust immune response or a more robust antibody response. But what we really wanted to ask is, what were we increasing the antibody diversity in these animals? And so why is that important? Well, if you're just producing antibodies that just bind to one single epitope within this CSP, as these bugs mutate or they undergo genetic drift, and if this epitope mutates, then you lose the immunity that you've actually developed to these bugs. So you want antibodies that bind at multiple sites within your antigen. So what we did is we generated a peptide array. These were 20 amino acid peptides that spanned the entire CSP peptidome. And we formed an immunoblot assay where we were able to determine which epitopes the antibodies our vaccine produced were binding to. So to determine what epitopes within CSP we were inducing an immune response to. So here's a heat map for those results where each one of these blocks represents a peptide epitope. What you can appreciate if you look at the results here at the bottom, where red indicates an increased antibody binding, you can see that the CSP pMan-TLR7 produces a more diverse antibody repertoire that bind at a greater magnitude to various epitopes within the entire length of CSP, and specifically to the N-terminal region of CSP. And the reason why it's important is because antibody responses to that region have been correlated with protection in children. So next we asked, are we actually producing a functional response? So here, what we wanted to do was, does the serum from our vaccinated animals actually inhibit the ability of sporozoites, the bugs that cause malaria, from invading the liver? So what we did is we isolated sporozoites from malaria-infected mosquitoes, and then we incubated them with the serum from our vaccinated animals, allowing time for these antibodies to bind to these sporocytes. And then these sporocytes are then incubated with primary human hepatocytes. And what we do is we then count the sporocytes that have invaded these hepatocytes. And of course, if our antibody cocktail that we've generated in these animals is quite efficacious, it'll be able to inhibit these sporocytes from invading these primary human hepatocytes. So looking at our results here, where the white triangles represents the serum from animals that were treated with saline. So here we have approximately 2 sporozytes per hepatocyte in these treated animals. The green represents the serum, the results for serum from animals treated with CSP plus the ASO1E formulation. And you can see that 1% serum concentration, we do see a significant inhibition of sporozoite invasion. However, you can see that the serum from animals that were treated with CSP plus PMAN TLR7 have a more significant decrease in sporocyte evasion at a 1% serum concentration. And that serum is even still efficacious at inhibiting sporocyte invasion at a 0.2% serum concentration, indicating an increased efficacy of the serum, of the antibody repertoire within the serum of animals that were treated with CSP PMAN TLR7 as compared to CSP plus the current state of the art. So based on the strength of our cellular and humoral immune responses we generated in mice, we have undergone a non-human primate study working with Sudhir Kasturi at the Emory Vaccine Center. And this is an ongoing study where we're immunizing animals with CSP conjugated to PMAN TLR7 or CSP plus the current state-of-the-art vaccine, and measuring things like T-cell activation as well as the humoral immune response for extended periods of time. And these are ongoing studies. So in addition to looking at infectious diseases, we've also turned our attention towards thinking about a cancer vaccine and using this as an adjuvant to adjuvant the tumor microenvironment to actually increase the immune response to tumors. So what we've generated is conjugates between our PMAN TLR7 and antibodies that specifically bind to tumor cells. What we do is we perform an intratumoral injection, and we've shown that these conjugates are actually localized to the tumor microenvironment. And then after we inject these vaccines over periods of time, we look at things like tumor volume and survival, and we're looking here at the tumor vaccine by itself, as well as in concert with other immune checkpoint inhibitors. So the green lines in this tumor volume study actually represent our conjugate system plus checkpoint inhibition, and the red line just the tumor localizing adjuvant by itself. And you can see, we see a very significant decrease in tumor growth, as well as increased survival in the conjugate system with and without checkpoint inhibition. And this is just showing that this is, in fact, this effect is mediated by Cd8 T cells, because if we knock out Cd8 T cells in these animals, we actually lose our ability to control tumor volume and increase the survival in these animals in Cd8 T cell deficient mice. So with that, I'd just like to wrap up my talk, and in conclusion, I've shown you a novel glycoadjuvant delivery vehicle that we can deliver antigens to dendritic cells or localize to the tumor microenvironment to induce antigen-specific immune responses. I've shown that we are actually able to target antigens to dendritic cells as well as activate those dendritic cells. And by doing that, we're able to induce a very high quality T-cell response, as well as a very diverse and efficacious antibody repertoire that is, in an in vitro model, able to actually protect against sporozoite invasion into human hepatocytes. And with that, I'd just like to acknowledge the others that have participated in these studies. Many of these studies were done during my postdoc at the University of Chicago and EPFL in Switzerland, working with Dr. Sachko Hirosui, as well as Michael Ranchi, Martina Damo, and Tiffany Marshall. I would also like to recognize my lab at Johns Hopkins University, who have been working very hard and diligent during this difficult, challenging time, and producing results for our next generation vaccine platforms that we'll be producing in the lab. So with that, I'd just like to open it up to any questions that anyone might have for this work.
Hi, Dr. Wilson. I was wondering what you would say is the main limitation that's preventing a lot of these immunotherapy vaccines from reaching a transitional or clinical stage and entering into the market?
Yeah, so that's a very good question. So sometimes it can be toxicity. So a lot of things that work in animals, a lot of these adjuvants, experimental adjuvants that are used in animals actually can be and are quite toxic in humans. So for us as biomaterial scientists, that should make our mouths water, because that's indicative of a delivery issue. So which are things that we like to specialize in. Another issue is the difference between human and mice immune systems. So the mouse immune system is actually quite different than human immunity. And when I first started to learn about immunology as a postdoc, I was initially reading a lot of mice papers. And then about halfway through my postdoc, I started learning more about human immunology, and I didn't recognize the names of any of the receptors or any of the molecules. They had completely different names. And that's because the mouse and human immune system are quite different. So oftentimes things that work very well in mice and look very promising just do not work as well in humans, or even fail at the level of a non-human primate. And to overcome that, there's a lot of work. For instance, Dr. Kim's work, he's starting to focus on looking at human immune system on a chip type systems, where we could, analyze these biomaterial-based platforms and other vaccine formulations and in humanized in vitro models, as well as there more and more each year, there are mice that have been genetically altered to have humanized immune systems. And so we can study these vaccines in those mice as they are invented and they're understood and they become available.
So many parallels to the talk I'm about to give. Actually, I'm going to be talking about humanized mice. I have a question for you, Scott, just in a general sense. And I definitely really appreciate the background that you laid the groundwork nicely in this first talk. So there's certain things I don't have to go into in so much detail, but you really beautifully distinguish the differences between humoral immunity and cellular immunity. And I know that generally speaking, whenever you activate, and one of the many reasons why cellular immunity can be toxic, especially with CAR T-cell therapies, is because when you induce a cell type like T-cells that can directly kill target cells, you oftentimes may have side effects even against host tissues. Now, in the model that you published in 2019, you showed this very marked, significant increase in Cd8 T-cells. Did you test whether or not all of those Cd8 T-cells were truly activated in an antigen-specific manner, or maybe there were others that were responding to IL-2 or other cytokines being secreted to just have a more general, broader Cd8 T-cell response, and whether or not you think long-term that could be a possible source of host tissue damage?
Sure. That's a good question. No, we didn't actually look at the antigen specificity of any of the T-cell responses in that study. We just monitored the antigen-specific response to ovalbumin and CSP and the other antigens that we looked at. But no, certainly, things like autoimmunity in response to vaccination is a very important issue. And I think it's about 5% of patients can develop some level of transient, usually transient, autoimmunity upon vaccination. So that also is a much larger issue. Unlike CAR T cells, which when you inject them, they're on and they stay on. The immune response to vaccination is usually transient, where you get this, as you know, Josh, you get this effector stage, which is a very robust immune response. And then that effector phase dies down, and then you develop a memory immune response. Of course, this thing like the bystander activation via IL-2, that's more primarily, due to the initial burst immune response that you get from these vaccinations. So, I think the solution there is higher specificity and better localization of immune responses. And then, of course, if you could bypass that initial effector stage and go directly to a memory stage, of course, that would be the best vaccine you could possibly imagine as far as thinking about infectious diseases and prophylactic vaccination. And we do have some interest in that, and that is starting to become a thing, is just going directly to memory stage T-cell activation would be the most ideal for safety, especially with children and the elderly. But that's a very good question.
And it's not entirely a fair comparison per se, because CAR T usually they deplete the host T cells to make them much more active without repressive mechanisms. So normally with CD8 response, you usually have peripheral tolerance and Treg inhibition. So that's another concern. Do you think when you're increasing these CD8s to such a large degree, are you also seeing increases in Treg subsets to potentially shut them down?
Yes, so that's a good question. The only thing we've looked at is in cancer, of course, in our cancer studies. We've looked at T regulatory cells within the tumor microenvironment. So we haven't looked at those within the draining lymph nodes of the spleen, so it's a systemic response. But certainly if you have a vaccine, you don't want to induce a lot of T regulatory cells within the tumor. So we aren't seeing the amplification of T regulatory cells within the tumor, but we also aren't seeing a depletion of T regulatory cells in the tumor. So that's kind of incomplete evidence, but at least we're not having a deleterious effect there on T regulatory cells.
No, but it's awesome work. I actually think if you look at the VDJ recombination possibilities, the amount of T cell receptor combinations is actually, I think, 5 orders of magnitude higher than what's possible with B cell antibody recombinations. The fact that not many people are looking at cellular immunity and vaccines to induce very targeted cellular immunity, it's definitely underused space of vaccination.
Yeah, absolutely. I think just getting something that induces a safe immune response in a human is already such a challenge that, you know, that's where a lot of the focus goes into. But certainly vaccine safety needs to be on everyone's mind as we design these things and move forward. You're exactly right.
Thanks for the great talk. Sorry, guys.
I had a little bit of a question about kind of the polymer itself. How does it actually matter? Or I guess the length of the polymer or kind of the average molecular weight of it? Maybe it's kind of more effective the longer it is or the shorter it is, or is it like a standard molecular weight to use every single time?
So that's a really good question. So we didn't do any very meticulous studies as far as on molecular weight. We did do studies on the composition of these polymers. So, you know, the monomer composition, because we had 2-3 different monomers and varying those monomer compositions. So as far as molecular weight goes, we were just trying to generate the largest polymers that we could because we felt that more was better. And the reason why is because we also wanted to limit the attachment sites on the antigen itself. And there were various reasons why we hypothesized that that would be important. We had developed this self-immolative linkage that was designed to come off, but it still has to come off. And the more of them you have on the antigen, the more have to come off. So that could be a limiting factor. And we wanted to deliver as much TLR7 per antigen as we could. So we wanted them to be quite long. But what we found was that by, as far as composition wise, increasing the TLR7 increased the efficacy of the polymer or its immune stimulatory capabilities. However, there is a limit, because if we got TLR7 levels that were above basically 20 monomer mole % within the polymers became insoluble. And we wanted to maintain their water solubility. So that was the limiting factor study that we've done on the polymer. And we also manipulated the amount of mannose as well. But we saw that the amount of mannose incorporated into the polymer had much less effect than the TLR7 on its immunogenicity.
All right, that makes sense. Thank you.
So there's a question in the chat. Which viruses are most known to provoke autoimmunity and promising candidates for a vaccine? So that's an interesting question as far as virus-induced autoimmunity. That's something actually I don't know much about. I do know, of course, viruses can cause cancer. You know, thinking about things like HPV, of course, that's a viral-mediated cancer. So I don't know as much about viruses causing autoimmunity. Now, certain vaccines can cause this transient autoimmunity, and that, as far as I know, I'm not an expert here, but as far as I know, that's more a function of the adjuvant and not the antigen. So the antigen carries the specificity for this particular infectious agent. But the non-specific immune response, which is generated by the adjuvant, is one more causing these autoimmunity issues. I don't know if that's an answer to your question, but if not, let us know. Okay, thank you.
Last call for any questions for Dr. Wilson. All right, if there are no further questions, can we get a virtual round of applause for him?
Thank you all very much. And thank you again for the opportunity to speak to you all.
Absolutely. Thank you again for being able to speak. It was a wonderful presentation, and I'm sure many students learned quite a bit. Yeah.
Hard to
So
follow that for sure.
as Dr. Doloff said, our next speaker is Dr. Joshua Doloff. Dr. Doloff is another prominent professor in the biomedical engineering department at Johns Hopkins.
Did not
With
think
his
that.
with his work having received multiple development and university provost awards. He's also been recognized as having the best doctoral dissertation in his graduating class with the Frank A. Bellameric Award. And during his postdoctoral studies, Dr. Doloff earned a Juvenile Diabetes Research Foundation Fellowship at MIT with the Langer and Anderson Labs, along with receiving several awards from various conferences and helping establish the Cambridge-based startup Sigilon. More than all of this, I have had the personal pleasure of knowing Dr. Doloff, as he is my own academic advisor, and I've heard nothing but good things from all of my friends who are in his lab or who have worked with him in the past. I have the highest respect for him, and it is truly my honor to introduce Dr. Joshua Doloff.
Thank you very much. Now I'm glad you are recording my talk for that part of it, actually. Thank you, Michael. That was very generous. So I guess I'll share my screen, and without further ado, I'll get to some very exciting topics that sort of follow actually in the wake of a number of the very exciting concepts that Dr. Wilson was bringing up in his own talk too. So just firstly, maybe you guys are working in nanoparticulate systems, maybe you're working in macroscale systems. So just as a quick confirmation, you guys can see my screen, right? Okay, great. So if you're working on the nanoscale or even on the low micron scale, so Scott just gave a very eloquent talk about nanoparticulate systems and how they get internalized by cells and how they interact at the cellular level. Right? And if an object exceeds the size of a cell, specifically increasing above around 5 to 10 microns, which is about the size of a cell, especially antigen presenting cells that can phagocytose an object. And so you start thinking about things that are micron or even millimeter scale, such as stents, little arrays for sensor arrays in the body, various implant systems, whether it's insulin pumps or continuous glucose monitors and pacemakers in the heart, bone screws, discs that you may put in the vertebra, and even large volume loss replacement engineered constructs too. So realistically speaking, Scott already brought up this concept that the reality matter is that the mouse immune system, we've even seen differences between different types of different strains of mice, and I'm going to be showing you some data on just that. Just in regard to the concept of foreign body response. So when you have an object that frustrates phagocytosis, it starts to exceed the size of a cell, you actually get multiple cells swarming the surface all in concert with one another. And I'll show you what that looks like. The macrophages, in this case, rather than dendritic cells specifically, fuse into these foreign body giant cells that try to really cover the entire implant surface. And that leads to ultimate sequestration by dense scar tissue that we call fibrotic or fibrous tissue or fibrous capsule. But in an effort really to understand better which strains of mice or which species of animals really translate well into humans, we also got into a concept, and I love this image online that I found. So this is the Vitruvian Man from Leonardo da Vinci, and then I love this adaptation with Mickey Mouse from Disney too. But where we actually irradiate the bone marrow of mice and we put in a human immune system so we can look at human immune cell dynamics interacting with our device systems as well. Okay, so. But just briefly for background, if you've never heard of foreign body response or fibrosis before, this was a device that Bob loves to present. It's called the microchips device system, and it's about the size of a USB key. Sits just under your skin. And what it does is it receives wireless signals. And so just like with telecommunications, with cell phones, there's an approved wavelength range for cell phone communications. There now too is a wavelength range for medical devices. And obviously there are additional implications for wireless transmission of signals for data safety, and also so making sure someone doesn't hack your device, or at least get your control, get a copy of your data in a way. But this device platform is very exciting because you can wirelessly through the skin send a signal that sends an electrical impulse that melts off one of these gold nanowell covers, melting the gold nano cover. And so any drug contained underneath that reservoir is released. And this was done repeatedly week after week. And this particular device came out of a human patient. They published this research back in Science Translational Medicine in 2012, just around the time that I was sort of beginning or just starting in the lab. at MIT. And, I was very naive. I asked Bob, is this some sort of plastic sheath you put around the device to protect it from the body? And he said, he laughed and he said, no, it actually is the foreign body response. And so something that I've come to appreciate over the years. So from the back end side of this, it looks very globular, phlegm-like. When you get sick and you cough up phlegm, it has that whitish, yellowish appearance to it, largely due to the fact, yes, there's some cellular debris, but it largely is due to the fact they have a heavy presence of white blood cells, and they're called white blood cells because they definitely, and we've seen it from even infiltrating responses that you can even visualize under the skin, even from particulate injection schemes for vaccination response, you normally see like a white infiltration zone. But interestingly, Bob then talks and he says, well, if you look at the interface along the implant surface, and so the implant would have resided in the top left corner here, he says there's a capsule that forms, and this is the fibrous capsule. So H&E, he says, that you can see the capsule after many months. This is after 6 months coming out of humans. The bottom image is Mason's trichrome staining, which is specifically used for visualization of collagen of all of the different types of connective tissues, and collagen usually is seen in blue. And so obviously you can see the major constituents of this new capsule are in fact new extracellular matrix collagen-based deposition products. And then Bob also said, well, by 6 months, we see very little immune response. And so he therefore claimed that it was no longer very inflammatory. And that is definitely true. But what we appreciate now is that over time, as the capsule forms, two things happen. One thing is that as it becomes a thicker capsule, the immune system sort of almost no longer sees the device. And as a consequence, its response wanes and dissipates and goes down over time. And this is usually a process that starts, the decrease is after about maybe 2 to 3 months. But then the other thing that happens too is that if you guys like doing a rock wall climbing as an example, I always use the analogy of climber's rope. And so very, very well, even just from a physics standpoint, that a single strand is very weak, but many strands woven together dramatically increase tensile strength. And so there's a phenomena that occurs in the implant field. When single layers of fibrous tissue are forming, not much is going on. But when you get this very thick sheet occurring, you actually get a process of contracture or squeezing of the device. It's a problem with softer material systems such as breast implants, hydrogel systems, and so on. Obviously, in the case of metal, this stainless steel surface implant, it's not going to distort it into a different shape. But interestingly, what you do see, even at 6 months here, is that when you peel the surface back, it's obviously very smooth because most of the surface of the implant is smooth. But take a close look here. You see this sort of circular divot in the capsule itself? It's actually the mirror image of the contours of the implant surface, specifically because it's squeezing that implant and becoming form-fitting. Okay, so Scott eloquently also went through a lot of molecular pathways in terms of cell activation. So the concept of foreign body response is not, I would say, quite as well understood, for instance, as toll-like receptor activation. But what we do know is that the immune system comes in, and whether it's naive cells in the tissue acting as immediate sentries, or it is cells that later get recruited in also to further potentiate this response. It's probably a mixture of both, we believe. The implant does not have self-recognition receptors for one, but two, there is also the phenomena and the theory that deposition by complement protein or other protein deposition on the surface of that implant increases the binding ability of immune cells to these objects. And so one example is antibody binding for opsonization too. I'll show a schematic on that a little bit later. But what we know is that shortly following implantation, even if you put the implant very far away from the incision site, you get this very large wave of immunologic infiltration. So all these little purple punctate dots, those are immune cells riding into that tissue. And you can see that they are very dense, smaller in size usually, and they're completely changing the molecular and the cellular landscape of what the tissue looks like. Now, the beauty of working with hydrogel systems, which we have for years, largely for transplantation purposes, for cell encapsulation as an example, it's a selectively permeable material. So you can have nutrients come in, oxygen come in, glucose can come in if you're doing islet transplantation for diabetes, insulin can go out to treat the recipient host. But in this case, I was really trying to just get a better understanding for the basic immune response occurring against the material. And you can see that in some cases it's non-fibrous, which we call quote unquote clean. There's very slight fibrosis, more complete fibrosis on these capsules. And then the fibrotic plaques actually start extending beyond the surface of the devices to the point where you even get extensive clumping, material sphere to material sphere, or to even other surrounding tissues, especially fat tissues. And so these capsules for scale, just because I mentioned scaling before, they're 500 microns in diameter. Okay, so much larger than the 5 to 10 microns. of an individual cell. And so early on when I joined the lab, we knew already that there was adhesion to the surface, plaque formation. We knew that it was sort of, with phase contrast imaging, you can see this very clearly on otherwise translucent materials. And then the engineers in the lab were starting to just do basic DAPI staining to know that it wasn't just protein on the surface, that there were actually live dynamic cells also on the surface of the object. Now, so whether or not you have a cell concept inside, so it's transplantation cells inside the material, we had lots of questions about which immune cells, were coming in, which ones were absolutely required. Were some cells coming in a manner that was just correlating response versus actually being causal for response? So with my systems biology background, I took it upon myself to start literally depleting different populations and perturbing different cytokines or chemokines to figure out which cells were absolutely required and crucial for foreign body response to occur. And we even found some kinetic data and then temporal sort of upstream versus downstream responder information too, which I'll show you. But this was the image that we had at the Koch Institute at MIT on display. And so there are islets in these capsules, but on the surface, you have this huge wave of green punctate macrophages. Obviously blue is DAPI for nuclei. The red is a surrogate marker for the fibroblasts that are largely responsible for extracellular matrix deposition. The marker is alpha-synuclein actin. But as cool of an image as that was, it's the worst case scenario. It looks beautiful, but it's a bad end result. So for my PhD, before I even got to MIT, I'd worked with a number of various animal models, skid mice, nude mice, non-skid gamma mice, black 6 mice. And so, you can sort of generally compare immune deficient versus immune competent status. But the problem with doing that across different genetic backbones of mice and different strains of mice is that you could also may be changing other things about that host animal that you're not fully appreciating. And so thankfully, when I came into my postdoc, I very much appreciated this particular point of doing cleaner science. And I realized that thankfully, not only the Black 6 model was the profibrotic model that everybody recognized, there were many individual single point mutations in that same genetic backbone where we could remove natural killer cells, T cells, B cells all by themselves, T cells and B cells together, all the way going down to hitting innate immunity on top of that. And then even doing targeted depletion studies, whether it was by antibody, by drug, or by liposomal formulation, where we could remove different immune cells. Now, just to summarize the findings, because it is a lot of data to go through otherwise, we did see about a 50 or 60% decrease in fibrotic deposition when you remove B cells alone. And I even identified the chemokine that links the recruits in B cells. It was implicated for B cell maturation in the lymphatics, such as the spleen, which Scott was talking about earlier, and that chemokine is CXCL13. So I used a neutralizing antibody against that, and the corresponding drop in fibrosis was quite similar. Now, in particular, though, when I focused in not on the adaptive immune system, which Scott was speaking mostly about, in addition to dendritic cells, which are technically part of the innate immune system, we really started to look into more of macrophage and neutrophils because that's what the literature sort of implicated before. I'll show you a schematic on that too. But only in the case really where we had innate macrophage dysfunction, they weren't removed from the mice, but they were not acting properly, or we just directly depleted them and then eventually inhibited them by drug, we lost fibrosis completely. So to show you the implantation procedures, you can either do injections of these materials under the skin, or you could make an incision down the midline of the mouse, pipette them into the animal, suture them, or use wound clip, suture the IP wall, and then put a wound clip over the skin with some tissue glue. One of the other cool things that we also did, we adapted this surgery to also try to do intravital imaging. At first, we were talking about the concepts of, can you even put a glass cover slip or a glass portal on the animal? Normally, this is done on the cranium where the skull is very hard, right? So it's metallized. You can glue a glass slide essentially directly to the animal and not have them pull it off. But obviously on the belly side, they're going to be grooming themselves. They may peel it away. And so we had this compromise solution where we kept the mouse under nose cone anesthesia, and we opened the animal back up, placing the animal face down on an inverted scope where the glass stage was actually the glass cover slip. It acted in a dual manner. When we first started doing these experiments, we used firstly an animal model that has green fluorescent protein, GFP, expressed in the macrophage lineage specifically. And when we first put these capsules in, we couldn't even see them. Everything was just black in the sort of the spherical area where the capsule resided. So we later encapsulated infrared range quantum dots. So now we can actually visualize the surface and know whether or not a macrophage in this case was actually touching the implant. And so On the left-hand side, we were able to see macrophages coming out of nearby fat pads based on 2-photon microscopy in conjunction with this intravital imaging. But you see here that macrophages 1 day following implantation versus 7 days following implantation, there's a much larger wave of recruitment into the area around these capsules. So macrophage response was definitely increased dramatically. We used a second animal model where EGFP, extra green fluorescent protein, was also under control of a different promoter, but this time looking at B cells. And so as compared to a mock implant where we do the surgery, there are many immune cells around your abdomen. Normally they're important for preventing bacterial leakage out of the gut and checking for pathogen exposure. But in this case, you can see them literally streaming once again out of the nearby fat pad and right in between the capsules into the fibrotic plaque. And so I told you before, we were in the business of trying to figure out what was going on. There was this stagnancy in the field for 30 or 40 years, even before I hopped into this type of work. And from speaking with others, we realized that, there was a holdover. Some would work in certain strains of mice and say our biomaterial is not fibrose at all. Then they go into monkeys or into humans and it fails miserably, right? So per Scott's point of differences between mouse and human. So what we realized anecdotally from speaking with others and then even testing this ourselves is that certain animal mouse strains or even animal models, such as other types of rats, had very low fibrotic index, and whereas other ones were very highly fibrotic. And part of this is historically, so Scott was mentioning the biopsy mice in his talk, they're known to have a very Th2 adaptive antibody heavy response, and that's great for vaccine efforts, which is exactly what Scott was talking about. However, in the case of foreign body response, we actually know that it's not antibody response that really drives this phenomenon. It's actually innate immune heavy Th1 immunity, which the black 6 mouse has in spades. And so thankfully, we learned that it translates very well up into the non-human primate. We did some work in cynomolgus monkeys at MIT. And so this is actually in the abdomen. We put these 500 micron capsules at day 0. This is minimally invasive laparoscopic surgery. So the cool thing of these studies was we can put materials in, and at later time points, we can go back in with a camera and take an image or take a video or actually take some samples or tissue back out. And in this case, what you're looking at is so in the mouse, if you open up a mouse, if you've ever worked with mice, you just see the anatomy like you would in an anatomy textbook. It's the GI tract coming down from the stomach, going all the way down to the bladder and then exiting the body. Well, you might have heard of the concept that fat is important for cushioning your organs. In humans and in monkeys, it's definitely the case. And there's actually this whole separate structure called the omentum. It's almost like a tube, a cylindrical tube of fat that is highly vascularized and that actually surrounds the entire GI tract, running all the way down. So the bottom of this image here would be where the lobes of the liver are. You look straight down the side wall of the omentum, all the way down to the very bottom of the abdomen, inside the IP space, the intraperitoneal space. And this lower level is called the Douglas space at the very bottom. And just 4 weeks later, you can already see that the capsules you start to lose resolution as they're getting embedded into the highly vascularized fat. Most of the time when we see biomaterials getting stuck to tissues, it usually is they're not primary organs such as the lobes of the liver, the kidneys, the spleen. All of those other primary organs have a very thick collagen capsule around them. You've actually seen where capsules get embedded into the lobes of the liver, like indented. We can pluck them back out and they're not fibrose at all. But in the case of the fat, it lacks higher compartmentalization and it has a lot of vessel density, and therefore you would imagine blood vessels open up, vasodilation, the immune cells move out from the blood, coming from the bone marrow, from other areas, and now they can swarm the surface of the device. Now, normally, when you look at fat tissues and you do sectioning or staining, so again, on the left is H&E for cellular infiltration, and on the right is Mason's trichrome. In the case where we have no implant, you see normal fat lattice. It looks like a spider web, or maybe like the open-bodied cells of cells in a cork, an image of a cork. But really, and why is that? Well, adipose cells are 50 to 60 microns. They're quite large, and most of what fills them is a lipid vacuole, sort of like a big water vacuole occurs in plant cells. And so when you section through them and you wash the sample to stain it and to incubate it, a lot of the lipid constituents get washed away. And so you're left with this outer membrane where the nucleus actually gets compressed all the way into the outer membrane because that lipid vacuole was so dense inside the cells. And over just even 2 weeks, you see this, but 4 weeks here, we show you that the capsules are now physically embedded in the surface of this tissue, and the tissue otherwise looks nothing like it once did. You have very dense infiltration. You can see actually red vibrant cells right around the surface of the implants, attacking the surface of the implant, macrophages and fibroblasts. And behind those layers, you have very dense deposition of new blue collagen that is forming. If you were to zoom in and do confocal microscopy, you actually see macrophages, Cd68 standing macrophages in green right around the surface of the capsules. You also see alpha-synuclein once again for fibroblasts also right around the capsule surface. Scott thankfully also mentioned the ability to take whole tissues and dissociate them into viable single cell suspensions. I had done that originally in my PhD, and I adapted those techniques to be able to dissociate cells off of the surface of biomaterial implants as well. And so what you're looking at is a grid here showing you in the top two cases, the mock implant on the left versus implanted on the right for Black 6 mice, and in the bottom case for these cynomolgus non-human primate monkeys from mock to implant. And so CD11B is a marker for all myeloid cells, the majority of myeloid cells. I think from the bone marrow, at least 80% of the cells are positive for CD11B. And there are two prominent populations that we see, one of which is are neutrophils in blue, and the other one are co-standing for CD68, that macrophage marker again that you see here. So you see the largest dynamic responder in terms of composition are actually innate immune macrophages. When we do multiplex gene expression arrays looking subcutaneously, we also put materials in sub-Q because that was the traditional site of biocompatibility assessment, and compared them to the abdomen implantations as well. You see once again CD68 up in both cases. You also see, here's an example of differences between mouse and even monkey immunity. LY6G, which is a marker for neutrophils, does not exist in monkeys or humans. So we had to use the marker CD6B for neutrophils here. But we found colony-stimulating factor 1 receptor, that earlier chemokine CXCL13, incredibly upregulated. It's on a different scale here because the background was so low in the mock implant that it was within the background of the assay. So we truly don't know the full fold change difference. We just know that it's tremendously upregulated in the implanted case. So reviewer asked us, we actually used a drug to inhibit this receptor, and it actually went on to block fibrosis. Reviewers asked us, well, do you see it only on punctate macrophages that are swarming into the tissue, or do you see it also on these large fused foreign body giant cells? And we see it on both. You see the small punctate dots, and you see it also in these large fused bodies that are around the individual capsules that you can sort of see pulling away a little bit from the nearby tissue. So here's a cool video of the laparoscopic surgery. So we've over time found a number of different new biocompatibility technologies that were licensed by Sigilin that Michael mentioned. And so you can see the capsules this is after 6 months. So we found technologies that even synergize. And so you see here, we're going back in, one port is for the camera, one port is for serologic. And so if you spray this area with saline, you can then suck the saline back up and then take any free floating capsules with it. But in particular, what you notice specifically is that the capsules on the omental wall are just clinging there just by association, and they are definitely not fibrosed. They get washed away, and there are no capsules remaining once we rinse extensively. Okay, so taking a step back, we know that Black 6 mice are very profibrotic. This was also known in industry, not just academia. These on the left-hand side we found to be quite compliant. We know that when you go to higher order animals, and we've done testing in New Zealand white rabbits and pigs, as well as the cynomolgus monkeys on the left, others have looked at baboons and rhesus monkeys too. We know that they're profibrotic as well. These translate very well into humans. There's always a little bit of a difference, other types of anatomical differences, but we ask the question, so we have this information where we know Black 6 seems to translate well. The molecular biology was quite preserved from the mice into the monkeys. And a drug target we found in the mice was useful later in the monkeys too. We did a long-term release formulation for that drug. But the question remains, there are differences between mouse and human. So can we, instead of getting monkeys all the time, which are quite prohibitive and costly, can you instead use a humanized mouse model? And so just very briefly, this is just a table, and I love the title of this review. It says of mice and not men. So this is just a table back in 2004 summarizing all of the potential differences, comparing on the left hand in the second column to the third column, mouse versus human differences. And you can see that the toll-like receptors are definitely highlighted in this particular example, but as well as many other facets of innate as well as adaptive immunity. Don't want to harp on that too much. So when you're thinking about putting a human immune system in a mouse, then it gets quite complex. You're like, well, how do you really deliver the immune system to fully recapitulate the full complexity that you would see in a human? And there's a number of varieties that are done of animal models and humanized models that are used. And you can deliver normally, the main delivery system is hematopoietic stem cells that are normally intravenously infused, but you can also put them intrafemorally into the animal. Some cases you can deliver peripheral blood leukocytes or white blood cells also into the spleen, into the IP space, intraperitoneally, as well as in the tail vein. And then there's another higher level complexity too, where others have started to take human fetal tissues, and so they're donated under a very regulated and controlled process. But the reason why they do this is because they also realize that human immune cells, or any immune cell, needs multiple tissue sites to truly differentiate and activate appropriately, not just the bone marrow that we're repopulating. They need the spleen, they need the thymus, they need the kidney, or potentially the liver. So fetal liver and thymal fragments are the most common. But so the immune cells can, at certain earlier stages of their development, they migrate there, they differentiate further, and then they move on to the next site. And so there's a whole nother, a higher dimensionality to these animal models. And so here's a summary of just humanized variants without fetal tissues. And here are ones with, and they're used for many different applications as an example, but here's another table, and from this really great review in 2019, talking about with the utilization of fetal tissue as well. In most cases, you're going to notice that most groups use non-SCID gamma mice, very immune compromised animals. And so I use them in my PhD for cancer studies, but they lack T and B cells. They have the SCID mutation, just like normal SCID mice, but they also have innate natural killer cell deficiency and macrophage and dendritic cell dysfunction. And the reason why most people go into heavily immune compromised animals to do this type of work is because when you have to deliver an immune system, it's very similar to bone marrow transplant. You have to first irradiate the mice. And if you have all of the immune cells present, you get a much stronger wave of inflammation, and it's usually much more toxic and life-threatening to the animal. So going to a more immune-depleted status to begin with, if you're going to remove it anyway, why not just have genetic knockout on top of that? It makes it easier. Well, so we irradiate. In some cases, you can deliver these fetal thymal tissues or not, but most times you'll at least impart the hematopoietic stem cells intravenously to get towards humanization. So we actually tried a couple of different variants of 2 or 3 different types of mouse models, and we did not see fibrosis. So black 6 mice, fibrosis on the left. Here is one version of the non-SCID gamma humanized system, no fibrosis with blank empty capsules. And then we even threw high levels of pig cells inside, so xenogenic, very foreign species, still no fibrosis. And what we realized from the work that I'd done in the wild type systems is that we were lacking and missing some basic constituents of the innate immune system. Interestingly, as opposed to the SCID mice where the adaptive immune system was depleted. So again, if I go back there real quick, so the adaptive immune system is gone, but the whole innate immune system is here. What we realized in these humanized variants was we were getting great engraftment of T and B cells. They were proliferating and being maintained, but we saw very low to no numbers of the whole innate immune system. And I've already told you that fibrosis is very innate immune dependent. So we went back, we modified this system further by adding in 3 human cytokines transgenically, and boom, lo and behold, we now get very prominent fibrosis with and without and with grafts inside the capsules. Now, what was also very interesting about this system, and so we have without cytokines in blue versus the full engraftment with cytokines in red, is that the percentage of CD45 cells, so general white blood cells, didn't really change much from the blood to the spleen to the bone marrow. But interestingly, when you wash out the immune cells, the PEC stands for peritoneal exudate cells, or IP lavage. You wash out the abdomen and you suck those cells back out and you analyze them. We saw very low migration of white blood cells to that space. And then with engraftment, it fixed this issue. But beyond that, regardless of CD45 status, we also did co-staining for CD33. In humans, this stains for myeloid cells. And in all of these compartments, we saw dramatic increases and maintenance of the innate immune arm of the whole immune system of these mice. When you look in the IP space, again, so there, again, increase of CD45, spleen, not a significant difference, but again, myeloid cells, it is a huge increase, and also peritoneal exudate normalized. So this is percentages by composition. This on the bottom, we actually normalized to total cell counts, cell numbers, and you can see that the differences became even larger in some cases for the upregulation on an absolute cell number scale. So just remember, when you're looking by flow, you have to be careful. Compositional data is great, but you also need, if you have 10% of only 1,000 cells versus 1% of 1,000,000 cells, you may actually have more cells on an absolute scale within that 1,000,000 cell population. So percentages are important, but not the full story. Okay, so in these humanized mice, we reconfirmed by macrophage depletion that macrophages were absolutely required, just like they were in the wild type systems. You got no fibrosis here on the right-hand side. But in addition, when we looked at the success of the engraftment, so one of the things that can limit humanized models is when you irradiate them, you may only partially irradiate the mouse bone marrow and the immunity. And so maybe you only get 80% removal or 50% removal. So we were really screening for, well, is most of the response what we're seeing due to the human immune system, or is some of the mouse immune system coming in to help as well? And so all cases here are normalizing the cell numbers. But for green, this is the numbers of CD45 myeloid CD33 cells after 2 weeks versus 4 weeks. It goes up on the surface of the capsules. When we deplete macrophages, it goes down at 2 weeks in red. Neutrophil numbers didn't really change much, CD66B again. When we look at the peritoneal cells, not on the capsule surface, but in just the general IP space, again, we depleted them very successfully around that sort of microenvironment. Neutrophils didn't change much, as you'd expect. The depletion was very targeted. When we look at mouse immune cells instead, so we use CD11B for myeloid cell identity with F480, which is a macrophage marker, very little presence in the tens, if not hundreds range. Same thing also for mouse neutrophils. They really were not abundantly expressed or present in these tissues either. So we know that what we're seeing is definitely truly specific to the human immune system. And what was also interesting on top of that is that when we did multiplex gene expression analysis, looking at completely non-engrafted non-Hodgkin gamma as just a low background control, and then we looked at engraftment with human immune cell addition 1 day later, 5 days later, and then 14 days later, many of these genes increase dramatically. And so if you have all capital letters, normally that indicates human. When you have only the first letter capitalized and the rest of the letters lowercase, that usually indicates mouse or rodent. And so human immune system specific probes were designed in a nanostring kit to look at these factors. And once again, we get human CD68 here. We didn't see human collagen 1A1, as you might expect, though, because in the human system, we're only delivering the immune cells and not human fibroblasts. But what was very interesting is that we had in the mice a slow, we didn't see them on day 1, but we did see them by day 5 and day 14. There was a slightly delayed kinetic, which we see in the wild type systems too, of later recruitment of mouse fibroblasts to come in. And we see mouse collagen 1A1 and mouse alpha-synuclein actin. So we know in this system, we're actually getting crosstalk between the human immune cells and the mouse fibroblasts. So really cool. Subcutaneously, the fibrosis was regained, and we actually see very, very similar effects subcutaneously in these mice. And what we know, I did staining for CD33, which is a human myeloid marker, and also for CD68. And what we're seeing here is the vast majority of the myeloid cells around the surface of these capsules are, in fact, innate immune macrophages with CD68 positive staining. We also see foreign body giant cell formation. We see this fusion event, these large fusion events around the surface of the capsules, and this is an overlay stain. It's interesting here. You can almost see like the fibroblasts are coming along the inside periphery of the foreign body giant cells surrounding the device. One last thing I want to talk about very briefly, because I know I'm going a little bit over here, is that we at that time wanted to get some spatial information moving away from the surface of the implant. And there was this really new exciting technology, not looking at RNA transcripts with nanostring. So nanostring, usually you would have a nucleotide probe with a fluorescent barcode that would bind to each individual copy of mRNA of the same mRNA species. And so you can get absolute counts of mRNA using this technology platform, and they get linearized, and you can count them on the surface. Well, they took this technology and they adapted it specifically to be interfaceable with antibodies, which have a UV photocleavable linker, and they have a DNA oligo probe that a microfluidic system. So down the bottom here, you stain with this 30, it's a 30 merplex, so 30 antibodies can be used on these tissues. You have some with fluorophores to determine regions of interest. Then you ablate those regions with the UV light, and it decouples the oligos away from the antibodies in that location. Microfluidic system comes up and you pipette off those probes, and then you incubate them with the barcode, and you can count, you hybridize and count the total presence of antibody staining. So this is what this looked like. We had unstained samples in the top left. You can see that in this case, it wasn't hydrogel. We also tested other polymers such as polystyrene, and you can even see some of the soft polymers roll up on themselves when they get sectioned. So we use CD45 and CD68 to visualize different areas around the capsules, and then we drew selective zones more specifically with these white lines, and that's exactly where we had very tight control over where the UV light was used to decouple antibodies right in those locations along the periphery of the implant surface. In discussion with the company, something else that was also really cool, because I wanted to get functional data moving away from the surface of the implant, we actually realized in speaking with them in early beta stage testing that they could actually increase the diameter of a laser incrementally. So they said, oh yeah, we can go smaller laser, use a small laser and get a smaller inner ring, and then move it out layer by layer by layer. And so ultimately we did this, and we had 4 consecutive 15 micron concentric circle rings moving out away from the implant. And the data that we got from that is really cool. We actually have some probes that decrease in an exponential manner, other ones that decrease in a linear manner. And I was able to graph these looking at hydrogel alginate at 2 weeks. It's much less immunogenic. At 4 weeks, though, it catches up, and it looks very similar to even something like a more immunogenic polymer like polystyrene that has much higher levels at 2 weeks and 4 weeks. This is log scale. Could also visualize this data with a heat map showing very similar things. So muted response with hydroalginate at 2 weeks versus 4 weeks. It looks very similar to these other materials. So again, it's a slower ramp up, different kinetic peaks of response. But we ultimately got very nice spatial temporal data moving away from the implant surface. And that full data set, which is not here because it is tiered in multiple levels, it's going to be in the paper that we've prepared and hopefully will be coming out soon. So we've achieved development of a humanized fibrosis model, first instance to ever really recapitulate fibrosis against foreign bodies in these systems. Verified macrophage recruitment in this model, also confirmed by depletion that is just the same in wild type systems as it is in these humanized mice. We confirmed fibrotic deposition sub-Q as well as in the intraperitoneal space. Foreign body response was preserved in response to multiple material implants, hydrogels, and different types of polymers. We even use PDMS, which is relevant for breast implantation as an example. Confirmed core cytokine signaling and immune cell kinetics, and many responses seen in humanized mice mirror those in Black 6 as well as the monkeys. Spatial confocal analysis shows quantification of immune response as a function of distance, which I promise you will see very soon when the paper gets published. I'm going to stop there. Sorry, I ran over slightly. And I take any questions if you guys have them. So it was a little bit more innate immune heavy, right? We realized actually that macrophages, so neutrophils actually do come in, but when I depleted neutrophils, the whole macrophage arm and fibrosis arm with fibroblasts still occurs. So originally the field thought the neutrophils come in first with protein deposition on the surface because they usually seem to be a very fast responder. But what we realized was they were occurring at exactly the same time. They were both there at day 1 following implantation, but neutrophils usually are responsive to, like, say, parasitic infection, like Scott showed with malaria contamination, malaria-borne parasites in the body. And so the neutrophils they spiked a little bit and then were maintained and then they went away over time. But they were not crucial. In fact, actually, just like there are Tregs which suppress overreaching immune response, there's a subset of granulocyte or neutrophil called a myeloid-derived suppressor cell that also has very similar negative feedback loop phenotypes. When we depleted neutrophils, the fibrosis, if anything, looked like it got actually slightly worse because the capsules clumped much more aggressively, which you don't see until much later time points. So it's peripherally involved in signaling, but it is not crucial for fibrosis. Yeah, we use this platform now. We're hoping that this humanized model can be used as a better platform for translation into the clinic. So someone just asked, JD just asked, can you comment on the ethics of using neonatal tissue in humanized mice? Yes, of course. So from speaking with our collaborators, obviously this is a point of debate in the field. Obviously, sometimes you publish papers with these systems and you might get angry letters from religious groups and different churches around the country, right? So, and that was told to me. But if you think about this concept of, well, if the tissues are extra to a baby being born, or maybe something happens and there's a miscarriage, right? So we're not necessarily taking tissues from abortions, as far as I know. So it's a very so all protocols, whether you're dealing with mouse animal models or human tissues specifically. So there's IACUC protocol committees for animal care and use committees. And then there's IRB, institutional review boards, that have to approve anything that comes in contact with human tissue, getting it from a human, and implementation of those cells or tissues. And so even just taking hematopoietic stem cells is a human cell type, right? We'd have to go through IRB approval just for that. So we collaborated with a group that already had access to a very specific collection mechanism that was about as morally ethical. It wasn't like, black market deals falling off the back of a truck. They were actually specifically going through very, very strong vetting processes to make sure the tissues were donated in an appropriate manner. There's full documentation for that, and that that they were being retrieved from procedures that were not contentious, obviously, and complicated in the modern day, which of course that whole area is complicated in of itself. Actually, if you look at stem cell research, when, for instance, it's very politically aligned. When George W. Bush came into office after Bill Clinton approved creation of all these different stem cell lines and lineages. And then when George W. Bush came into office, he put a moratorium and said, okay, all the things that were created, that's all you have right now. You can't make any new ones. And so it is obviously based on the way you're born and the things that you believe. Yeah, it's an area of research that you don't have to work in. Actually, a question that I have is, there are all these humanized variants that you don't even need neonatal fetal tissue. And can we better engineer those systems specifically to be able to recapitulate this multi-site requirement for proper immune cell development and differentiation? And that could be something that you take on. You have to obviously compare them to the fetal cell systems and then show they work just as well before you completely move away from those. So I think it is definitely something that we can do better with. And it's another reason why people are moving away from embryonic stem cells towards induced pluripotency, also because that in and of itself, it's a renewable cell source. You don't have to get grafts or tissues from any donor, right, or any animal. You don't have to worry about sacrificing life in order to improve or to make life better. And as of right now, most organ donor lists, as you know, are quite long. Organs are limiting for sure. You're welcome. You're welcome, JD. I know I'm over a bit, so we can edit there if you want, unless anyone else has other questions.
I had one quick question.
Sure, go for it, Alex.
Because I know you brought up induced pluripotent stem cells. Is there like any specific reason or mechanism by which neonatal stem cells work better than induced pluripotent stem cells?
Well, don't think about it so much that it's the stage of where you're getting the cell from. It's actually more important to have the hematopoietic stem cells, you can get them from adult, you can get them from earlier stages of development too. We know from cases of Dolly, the sheep being cloned, that the older the cells are, potentially the higher incidence of other diseases later in that animal's life, they can arise sooner in their life, because it's almost like starting their telomeric biologic clock at a much later point in time. To me, it's not so much about needing neonatal per se, it's more about having the actual organoid tissue, the 3D structure and the 3D, like the layers of, for instance, the lymphatic tissue, like the spleen, in order to allow immune cells to move into the germinal centers or into the right locations to be able to be presented with what they need to be presented with, to mature in the way that they need to mature. And so so now like Encore's saying at Georgia Tech and many others, like they're using biomaterial systems to literally build whole organoid level structures. And so hopefully when that field gets better and we can start implementing those systems in animals, maybe we will no longer need the fetal, think about it from the fetal tissue rather than the individual cells. It's more about the tissue microenvironment that's needed.
Thank you.
Sure. All right, guys. Well, if that's it, thank you, Michael, and to the rest of the committee for inviting me here today. I very much appreciate meeting everyone. And I look forward to the rest of the talks. Otherwise, enjoy the rest of the event, guys. Thanks again for listening.
Thank you so much, Dr. Doloff. If we could get one more virtual round of applause for him. All right. Our next speaker is Ilon Heylesius. Ilon is a developmental engineer at Stryker focused on developing solutions for degenerative spinal cord conditions. As one of the global leaders in developing medtech, Ilon has survived one of the toughest master's programs in the world and is an inspiring success, especially for many of our current and past students at Johns Hopkins. A previous recipient of the Alliance for a Healthier World grant and a prior design studio teaching assistant for the biomedical engineering department, Ilon is definitely someone to look up to as a role model of what many of us students aspire to be. My close friends have the highest respect for Ilon as both a scientist and individual, and it is my pleasure to introduce Ilon Heylesius.
Thank you so much, Michael. And thanks for inviting me as well. Just sharing my screen here. Do you see my first page?
Yep, looks all good.
Okay, cool. So hello, everyone, and thanks for having me here. It's really exciting to be just listening in on Dr. Wilson and Dr. Doloff and just the research that has been done, and then also just seeing how interesting the conversations went with the questions and how excited you guys are to just be here. And I just wanted to, I was asked to speak today about my current work at Stryker, a broad overview of my R&D experiences from undergraduate to the workforce. So I'm going to be touching about a little bit on each of these topics. So I just wanted to start off by going with my background. So I started off my undergrad at the University of Arkansas in Fayetteville, Arkansas, where my family was. When I was there, I studied biomedical engineering. I worked in a wet lab research group, and this was since my freshman year to my senior year. And the research was mainly on cardiology, and cardiac stem cells, and just using biomechanics to create blood-brain barriers and different types of research with those cardiac cells. I also was part of different groups, international culture team, Bible studies, missions trips. I did several study abroad and service abroads. And I put an image here of the Engineering World Health Chapter logo, because this is one that was close to my heart. My college wasn't really focused on, medical devices or, using mechanical engineering for medical devices. It was a lot of wet lab research and tissue engineering. And so I was very interested just in the medical device aspect of things. And this is why my classmate and I, we decided to start Engineering World Health Chapter at our university. And a lot of students were interested in it. And it's really cool because it's continuing to go on right now at our university. And it was a great way for me to get involved in the medical device industry with students from different universities as well. I was part of the BME Society and different parts of the engineering department and design classes and everything. But another experience that I really enjoyed in Arkansas was my Amgen Scholars Program that I did at Harvard University after my junior year. And when I was there, I was working in George Whiteside's lab. He was the founder of, paper-based medical devices. And so these are like diagnostic tools that we created from his lab. So it was really cool just being in a lab and with other Amgen scholars from all around the US and just being connected to that community. So I graduated in 2016, went on to an internship at the USAID in Washington, DC. And this was a global health internship, not really focused on engineering or the medical field. It was mainly global health and healthcare and international development, which was one of my passions, and I'll go into that a little bit later. So I had my engineering background at University of Arkansas, wanted to go into understand a little bit more about global health and international development. And that's where I was able to spend time at USAID and interact with people all around the world and see how healthcare data management was taking place and how that policy was working with the government. From there, I finished in May 2017 and went on to Johns Hopkins' CBID program. I'm sure some of you may have heard of this program, the Center for Bioengineering, Innovation and Design. And while I was here, I joined a couple projects. I was put on on a team that worked on a project in the US and a project internationally. And we got to travel to different countries to perform usability studies and also just talk with surgeons in India, in Europe, in Africa, and also in the Johns Hopkins medical environment, just talking with surgeons and getting to know the innovation process through our engineering classes. And I was fortunate to take a computer-aided design class, which I'll touch about a little bit later. And it was an amazing experience at Hopkins. One of my projects was called Idea Medical, as you see here. We were able to continue on with that project. And in fact, right now it's considered a startup and has, this week we received news that we're moving into a new office in the Baltimore location. And so it's definitely a growing company, which is really cool to see how that stemmed out of the Hopkins engineering program and how students are continuing that project too. So graduated in June or May of 2018 and started at K2M. And K2M was a company that was focused on spinal instruments, spinal implants for surgeons. And about 5 months after I started, we were acquired by Stryker. And so I've been with Stryker since then. It's kind of been a mix of the company cultures. It's really cool to see that I was able to start at a small company, but then now have that large company feel and see how that transition worked out. And this quote at the bottom right here, I don't want to pass over it. It's been a family motto for my family. I grew up in a family of engineers, my parents and my sister and me. And this is just a quote that we always say at our table, whenever we talk, whenever we're working on projects or anything, we have to say, you know, if it's not me, and then who? If not now, and then when? There's always people waiting, and there's always time to do something. And if you're not acting on it, you know, when will you do it? So that's just something I wanted to share as well. So it may seem like I had each step stand out through my journey, but it didn't quite work that way. And I'd like to share a little bit more about how I got to where I was before going into the details of what I do each day at work and the background of the spine, how I work with the spine and with surgeons. So I just wanted to share a little bit more of my pathway, just to hopefully encourage you all as you're working through your college careers and thinking about your path ahead of you. So I grew up in Pennsylvania, and my parents had immigrated from Ethiopia to the USA. So I was living in two cultures. I grew up with listening and learning two languages and a whole bunch of different foods always in my house. Cooking and smelling all the Ethiopian food was always amazing. I grew up going to a private school, and I was able to learn a lot from my classmates and my teachers. And in 2003, I was in elementary school. I had an eye injury, and this is kind of like how I started my interest in being in the medical field. And I always thought I was going to be an eye doctor, because after this eye injury, I was just in shock of how well the doctors took care of me and how I was able to get through my injury. And so I pursued more science and engineering and math clubs through my library and through summer clubs and activities. And in 2006, a few years later, I got a chance to go to Ethiopia for the first time. And this is where my family is from. I was able to see their hometown, where they grew up, and then also go and see where my cousins were living and the healthcare system in their area. And I really saw the desperate need for medical devices and healthcare development in the country. And that's when it started me, you know, maybe I could also help just being a doctor and also like working on medical devices and helping with helping to improve the healthcare in this country and other countries around the world. So I continued through middle school and high school, joined different sports, varsity athlete, and trying to be just dedicated and build up my work ethic as well during high school. So I applied and was accepted to several of my top schools, including Johns Hopkins for my undergrad. And it was an interesting story, because I know I talked about going to University of Arkansas. So a lot of my classmates were even surprised about, you know, how I declined my offer at Johns Hopkins for my undergrad, because I was the first from my high school to ever be accepted to Johns Hopkins, being a private school. So it was kind of a tough situation for me. And I wanted to touch on that a little bit, because it all goes to priorities. So my family was moving to Arkansas. My dad had recently got a job in Arkansas, and it would be really difficult and challenging just to be up in the East Coast while my family was down in Arkansas. And just for different family reasons, we were able to just move our whole family down from Pennsylvania to Arkansas. And I knew in the back of my mind that I would either continue with med school or grad school at Hopkins, because I knew that was my dream school. And I'm really thankful that I had that assurance, just in my mind, that I would come back and someday it would work out. So in Arkansas is when I started my BME major, and I actually chose biomedical engineering as a backup to med school. I was thinking that, you know, just in case I didn't want to go into the med school field right away, or I wanted to just focus on my engineering, my math skills, because I loved that so much, I would choose BME major. And I'm so thankful that I did that, because after my sophomore year, I realized how much I really enjoyed making and designing and developing devices and just creating solutions for surgeons. And so I'm thankful for that decision. And that's kind of how I ended up on my journey. And you know, through my journey, I focused a lot on research that was not in the field that I'm in right now in Stryker. So I'm going to my next slide right here. Right now in Stryker, I focus on the spine. So this is, I'm sure you guys know, the vertebrae in the spine, all the way from the neck all the way to the pelvis. And this is something that was completely new to me because, as I mentioned before, I was working in cardiology, and then my master's program was focused in ophthalmology. And so going into K2M, now Stryker, it was a completely new field. So just wanted to encourage you guys too, if you're interested in any kind of field in the future, don't ever give up in pursuing what could be new, and don't worry about taking a risk. It was definitely risky for me to go into a field I didn't know, but I knew I would learn on the job. And that's exactly what my hiring manager and my coworkers all encouraged me as well before going into the spine field, because they saw, coming out of Hopkins, I'm sure a lot of you students from Hopkins or from other universities that are focused on research, and even just being here on a Saturday, that shows that you're dedicated to and committed to learn. So it's really important to build that work ethic, and then you learn on the job as well. So going into the spine, as you see, there's different vertebrae in the spine, and each level there is a disc. So this blue disc in the top right corner is the intervertebral disc. They're in between each level of the spine of the vertebral bodies, and the vertebral bodies are labeled. You can see L1 in the bottom right here, L1 to L5, and the thoracic section is T1 to T12, and then the cervical from C1 to C7. So in between each of these vertebrae are these discs that are colored blue here. And these discs act as shock absorbers. They're full of water, they're very hydrated, and they act as cushions in between the bones, so the bones don't rub against each other. And sometimes these discs get in the way. And there are some spinal conditions that are involved with these disc destructions. So if you see in the top right here, this is a normal disc where it's fully hydrated. The next disc right here is a degenerative disc. And so this is where it's called degenerative disc disease, where the disc just collapses on itself because it's dehydrated or it's just overused. So the name is kind of misleading. It's not really a disease, but it's a condition where there's a damaged spinal disc that causes pain. And the reason this causes pain is because it pinches on the nerve. And so if you see in the back of the vertebral body, there's an opening where the nerve comes out, the spinal cord, and there's nerves all around the disc, all around our spine. And so if the disc touches a little bit on this nerve, just very slightly, it can cause pain. And that nerve goes all around the body. So sometimes surgeons are able to detect what level the spine, what level the disc being affected based on where the patient's pain is. And so if a patient has pain in their left leg, the surgeon will know, oh, that's a L4, L5 disc, because they know where the nerves are leading through the body. Another spinal condition is called radiculopathy, and this is where there's a herniated disc. So the disc just slides backwards, and it's kind of the same issue. It impinges on a nerve. So this is the kind of situations that we are building devices for surgeons that treat these kind of cases. So at Stryker, these are the devices all from our company, from K2M and Stryker. So K2M just being acquired by Stryker. And so there's interbody cages. These are cages that are all of these are interbody cages. Basically, they go into the location where the disc was. So if you see in this picture right here, between the vertebrae, there is this white disc, this white cage. And so the cage is placed there where the disc used to be. So the disc is removed, all the pieces of the disc are removed, and then the cage is placed in there. And then sometimes there's a plate that covers it with screws, or there could be screws that are already embedded into the cage. And so the surgeon will simply need to just put the screws in the cage once they insert the implant into the body. And so there's allograft also. Allograft is the graft material that you can see that all these cages have holes in them. So the graft material is placed inside the cages so that the graft can grow, so that the bone can grow through the cage, so that it can grow bone to bone. So it's kind of like a fusion that happens. So I was going to show a short video about how these cages work, but since we're short on time, you can feel free to open up YouTube and search how these cages work and how they expand. It's really cool to see the science behind them. So in Stryker's spine, there's several different roles. The R&D engineering department where I'm in, the marketing, which talks directly to the field, to surgeons and to sales reps. So sales reps are in the field and they are the ones working with the surgeons. And then there's quality assurance, regulatory, sourcing, supply chain that talk directly with their manufacturers, and a patent team. So these are different roles, and these are filled by engineers, by business majors, by even material science majors. There's tons of different people that backgrounds that people have in these fields. So collaboration is definitely key in our field, as I'm sure all of you guys know in science is super important. So into my engineering role, I work a lot with surgeons. We interact with them. We have brainstorming sessions with our engineering coworkers and just with our advanced development teams. We use a lot of SolidWorks, which I'm thankful for that class I took during my Hopkins master's program. It's something that I encourage lots of students to take, especially if you want to go into the manufacturing field or a medical device. And this is it's important to have that cross-functional collaboration. Another interesting part of my job is I'm able to visit cases. So I've been able to travel across the US to visit when surgeons are actually using the devices that we create and actually placing them in patients to treat the patients. So that's been really cool. So my job focuses a lot on additive manufacturing, which is how those implants are created with titanium powder. I'm just going to play a little bit of this video that shows
titanium technology, which utilizes advanced 3D printing methods to create structures once impossible with traditional manufacturing techniques. Starting with a titanium powder, the implants are grown through the selective application of a high-energy laser beam. K2M's lamellar titanium design incorporates complex internal geometries and rough surface architecture, which has been associated with biologic activity. It has been well established that the surface properties of biomaterials have a significant impact on the biologic response to the in vivo presence of an implant. Titanium has a long history of usage in orthopedic applications, and recently there's been a great deal of research into surface modification techniques to further enhance the bone healing adjacent to those surfaces. K2M's lamellar titanium technology incorporates a 3 to 5 micron surface roughness, which has been shown in peer-reviewed research to increase osteoblastic activity compared to titanium and other biomaterials such as PEEK. Orthopedic implants have long incorporated porous structures to facilitate direct bony integration with the biomaterial to achieve enhanced fixation at the bone implant interface. A great deal of research has given an understanding that interconnected pores in the diametrical range of 300 to 700 microns are ideal for allowing bone ingrowth through porous biomaterials. K2M's Cascadia interbodies incorporate 500 micron diameter pores, which run through the walls of the implant from end plate to end plate and serve as a conduit for bone incorporation.
All right,
so
that's kind of what I wanted to share about
Introduce
the Cascadia lamellar technology, which is these implants. This is a system that we now have through Stryker. So as you saw in the video, it's the way that these devices are created are through the titanium powder that has been laser scanned and melted on a platform to build these kind of devices that have very unique features. And the surface roughness is definitely a selling point that we sell for surgeons, because surface roughness and these pores that go through the entire interbody cage are very important for bone growth, for those osteoblasts that build the bones together, and for the mesenchymal cells that come out of the bone marrow so that the bones can grow through these cages for the patients. So this is our mission. Just wanted to put this in here because Stryker is very driven to make healthcare better with these values. And I learned a lot through dedication and commitment. As I mentioned before, I was able to travel to different countries for during my undergrad and my master's. And one thing I learned when I was in Vietnam was the merchants, when they're there, they go early in the morning, they go to the river market to sell their produce and just to make income, and how dedicated they were. And that was just a lesson I learned from one of my trips, just being dedicated and committed to your work, and having that work ethic is super important. So I know we're running out of time, but these are just a couple more slides I had about Stryker's divisions. If you're interested in learning more, there's stryker.com, the website. There's also divisions all around the world. We work with people in Ireland, in India, South Africa, and Europe, and Latin America. And there's over 40,000 employees, so you can always get connected to somewhere that you're interested in. So one thing that I wanted to touch on before finishing is this program that Stryker works on and supports. It's called Operation Smile. And if you're interested, there is more to learn at this website, smile.stryker.com. Stryker is very big in giving back to the community, and they help out with these children that have cases of having these cleft palates and cleft lips. And Stryker is very supportive and even sends employees out to the field to different countries, to Morocco, Rwanda, Nicaragua, and several other countries to support these kind of surgeries. So just something that I wanted to touch on before we finish is just through college and through work, I've learned a ton of different advice and different tips to collaborate. And just something that I really want to share is encouraging you guys to get involved in volunteering and networking and just interacting with people even outside of your field. You might think that it might be different to talk to people who are not in your field, but it's really cool to get ideas and to share with people who have different backgrounds than you, different cultures, different upbringings. And it's something that really has made a huge impact on my life too. So if you want to learn a little bit more about my background or go into more details, I did do a podcast talk with Medtocracy. It's also a great group that they have several episodes out now on LinkedIn. You can find them. And this was started last summer. And then also there was a career blog that I was featured in August of last year. So thank you all for letting me have the chance to speak. And I hope you guys all enjoyed learning a little bit about my background, and also interested in the spine a little bit more. And I'd be glad to get in touch. My LinkedIn and Twitter, my first and last name, and my email address is right there as well. So hope you can reach out, and I'd be glad to share a little bit more and to learn a little bit more about you as well. So thank you.
I want to ask a quick question, if that's okay.
Yep.
So thank you for the talk. So from my understanding, the CVID master's program is heavily design team project based. I think a lot of the undergraduate engineering programs have some sort of equivalent. So I was wondering how your experience in CVID in an academia student setting with design differs from what you're doing now at the industry level.
That's a great question. Yeah, I have seen a lot of similarities. So in CVID, we spent the whole summer, basically all of June, July, and some of August, observing surgeries and talking to surgeons and interacting with surgeons about their needs and questioning them, what do you actually need in your surgeries? And that's kind of what we do at Stryker too. We bring surgeons in-house. We have a cadaver lab in our facility in Leesburg, Virginia. And we bring the surgeons in, and they use our devices, and we ask them, what do you like about this? What do you not like about this? What do you think is there anything that you would want to change about it? And then we go back to the drawing board, we go to SolidWorks, and we have a 3D printer, a plastic 3D printer in-house in Leesburg as well. So we are able to prototype and design kind of like we do in the design studio at CVID. So it's a lot of similar design processes, and the innovation process is very similar to learning how to use design history files, talking with the FDA, and that's what we learned in CVID, and that's exactly what we're applying in our real life. So it's very similar, and a lot of the managers at my company know about the CVID program, so they're very familiar with how that works, and they're excited whenever students apply from CVID, I'm sure. Any other questions? All right. Well, I have my LinkedIn and my Twitter and email. If you ever have any questions, feel free to reach out. I'm always available, and I love just talking with interested and dedicated students.
Thank you so much, Ilon. If we could get one more virtual round of applause.
Thank you.
Now, our next speaker and the final one before our intermission is AJ Bezub. Hailing from the Paul Corporation as a value stream leader, AJ is an alumnus of Johns Hopkins, and he's actually someone that previously occupied my current role, as he was the former president of the JHU SFB chapter and was integral in the founding of our chapter and starting the Mid-Atlantic Biomaterials Day conference as a whole. As someone that many of our students admire due to his success both in and out of Johns Hopkins, AJ was a natural leader who inspired others and continues to motivate our students today. Although I only personally knew him when he was graduating and leaving Hopkins, I can confidently say that AJ is an amazing individual, and it is my honor to introduce AJ Bezub.
All right, thank you for the introduction. I guess, can everyone hear me / see me? All right, good.
Yep.
So yeah, thank you for the introduction. Thank you for inviting me. Obviously, SFB does have a very special place in my heart because of, when I was back in Hopkins, me and I gotta say, James Shamel, he's at UMD right now. He was, kind of the, the brains behind Mid-Atlantic Biomaterials Day, and also, I can't really take most of the credit for this because it really was a lot of him. I was more of a supporting role, but yeah, thank you for the invite. I'm gonna go ahead and share my screen. So here we go. So can everybody see the slideshow?
Yep, looks good.
Alrighty, cool. Let me, from the beginning. Alrighty. So yeah, my presentation is not going to be overly technical. I know a lot of the talks and presentations usually are with SFB, but it's more of sort of a common sense sort of thing, like a business overview. I'll have a little agenda a little bit later, but from oil and gas underground to aerospace in the atmosphere and all the biomaterials in between. See what I did there? Anyways, Paul Corporation is a company that I didn't hear of until a career fair my senior year. Not a lot of people do know about it, but we provide filtration, separation, purification solutions for all applications under the sun. I'll get to that a little later. And since the beginning of my career, I've always worked in operations, so I've never really been on the technical side of things, but I still, am very passionate about all the different applications and such. So, I'll go into a bit of a business overview, a little bit about my journey with the company thus far, go into the Danaher business system. So Danaher is the parent company of Paul Corporation, and they also own companies like Cepheid, Leica Biosystems, Cytiva, which is the GE, was the GE portion, or the biopharm portion of GE Healthcare, Beckman Coulter. So Danaher is, I think they're number 120 something on the Fortune 500 list. Very, very big manufacturing conglomerate. And then going to go into a little bit of applications of some of our different products, and then obviously be open to questions at the end. So as I kind of hinted at before, the markets we serve at Paul are it's all of them. So mostly life sciences. I say that's about half of our business, maybe a little more, especially now because of how the COVID pandemic has impacted the world. So, biopharmaceuticals, I can read everything over there, but, in the medical industry, especially with, IVs and different applications, in that some of the products that I oversee at the manufacturing plant here at Pensacola in my current role go directly into, IV applications, water applications, ventilators there at the bottom, especially recently, that's been a big topic, and Pall Corporation actually got this is close to $5 million contract from the DoD back in August to kind of help bolster our manufacturing capability of ventilators at our site down in Puerto Rico. So we usually pump out about 480,000 ventilators a month out of that site, and that contract's going to help us to build new equipment and kind of expand our process capacity to make over 650,000 ventilators a month within like a 16-month time frame. So that part of the business is obviously expanding. Over in the food and beverage side of things as well, beer, wine and spirits. I have a little thing on the beer part later. It's kind of funny how we kind of have our hands in that industry as well, and how we provide sterile filtration solutions in that industry as opposed to the current practice which most breweries use of pasteurization. So as far as my experience in life sciences, I was at the site in Hauppauge, New York, up in Long Island for my first year with the company, and we made the filter media as well as the filter devices for tangential flow filtration systems. That's used in a lot of pharmaceutical applications, sold to all the big pharma companies. And, of course, I don't know too much of the technicalities, but the one thing about Pall Corporation is that in addition to having multiple industries that we serve, we also make the filter soup to nuts, right? So when I say filter media or filter membrane in the life sciences context, that's the raw material that goes into a filter. So think of it like as a giant roll of toilet paper, basically, and it varies in sizes and different slit widths depending on the filter you're making. And so that has the special chemistry, and the material science part is all in that filter media. And so we manufacture that on a very large scale for all different types of applications. Sometimes it's a supported membrane where we'll buy a substrate from some other vendor, and then we will do what we call cast membrane or a media on top of it. So some sort of chemical formulation, and then it goes through some environmental chamber or other chemical process, radiation if we're doing surface treatment, stuff like that, to kind of give that filter media the properties that make it do what it's got to do, whether that's ion exchange membranes or something like in venting applications where you're only allowing air through but not water, and you need like a certain water breakthrough pressure for different venting and IV applications, as well as in like autoclaving, stuff like that. And then back to my original point with the Hauppauge site, we made the filter media and then also the filter device. So after you make the media, you have these giant rolls of media, and then you have to make them into a device. So at Hauppauge in Long Island with the tangential flow filtration systems, it's just kind of like stacking the media in a certain size after cutting it to a specific size. Then you just stack it in a certain sequence, certain number of layers of media, and what we would call screen and permeate layers as well, made of different materials to kind of make the filter do what it's supposed to do, which is kind of capture particles of a certain size. So we would classify them in kilodaltons. So like a 50K filter would let everything 50 kilodaltons and below through the media, but anything above that it would capture. And a lot of the times you hear pore size with filters, whereas in reality, the filter media provides more of a tortuous path through which bigger particles cannot travel, and the smaller particles can. So it's like we might say pore size, but really it's the chemical formulation kind of making some particles get stuck and others not. So industrial markets served. So again, make the media and the devices for all these industries, especially in a lot of these, well, not a lot of the industries, but especially the aerospace industry with the pandemic has gone way downhill, unfortunately. I was, so after being at Hopog for a year, I was in Newport Richey for a year, which is a city north of Tampa, Florida, where we manufactured filters for aerospace applications. And, I was there when the COVID pandemic started in, February, March of 2020. And we, started laying off people. And, so that's one of the things about, working for the company is that you get to see the highs and lows of everything. And in June, I left Newport Richey, came to Pensacola. We're more life sciences based, and we can't hire enough people at this point. So, it's, just a side note there about serving all the different markets, but, also different process technologies, especially oil and gas. So upstream, where kind of you capture those hydrocarbons, and then you separate the oil, the gas, the water during the midstream applications, and then downstream before things enter the refinery, and you get into more the nitty-gritty of those, oil and gas processing, separating the contaminants and particulates that could damage your equipment while doing those more fine, refining applications in oil and gas. And if you're interested in learning more about any of the industries we serve, all of the information that I'm using in this presentation is straight off the website. In fact, the website has, much more detailed information than what I am regurgitating here. So, paul.com, and then it's got all the industries there. If you're interested in learning any more about any of these particular applications, with aerospace, kind of whatever, it's commercial, military, rotary wings, so helicopters, fixed wings, so, F-35s, Boeings, the rest of, traditional airplanes, also marine applications, and, not only helping to, filter out like cabin air filters and stuff, but also in hydraulic applications, lube applications, fuel, even avionics. So like the electronics equipment on board those vehicles, if you will, filtering the cooling air that cools the electronic systems to help them maintain their service life and reduce maintenance costs and upkeep costs. And then microelectronics is another very, very big industry for us, specifically the plant. We have a manufacturing plant in Cortland, New York, and upstate that does a lot of the applications for this. There's also a big R&D hub up there. So in semiconductors, electronic components, we sell to companies like Samsung and such when they're manufacturing phones and, the, silicon chips that go into phones and stuff. Basically, one of the grades of media that is manufactured on the line that I oversee in Pensacola actually removes contaminants from IPA before the IPA cleans like a silicon wafer. So it's like we're filtering the cleaning fluid for electronics, and we're just talking like nanometer scale, pore size, if you will, media. So it's very, very fine stuff. And I don't know too much of the details of the exact chemistry and material science in that particular application, but you kind of there's something for everyone in Paul Corporation is kind of the point of all the markets that we serve, and sharing these slides. So I'll go into a little bit of my journey. I know I've kind of already hinted at where I've been, and also I'm originally from Chesapeake. Obviously went to Hopkins. A couple fun facts there. I've heard Phonathon is still a thing. Students are working from home now, which is really, really cool. I absolutely love that place. I spent all 4 years at Phonathon. And so I started my career in Paul Corporation's Operations Leadership Program. So it's a program wherein they scoop up engineering students out of college, eager to work and spend their life in manufacturing plants. And you get 2 rotations, so they're a year long. So 1 year at one location. So my first year was in Hopog, New York, mostly life sciences. We produce filter media and filter devices. So it was cool because no other plant within Paul Corporation does both. Usually a plant is either a media site, so producing the raw material, or a device site. So I was a manufacturing engineer for 6 months, and then a DBSL for 6 months, so a Danaher Business Systems Leader. So basically, the title at most companies is like Continuous Improvement Manager. It's just the company Danaher calls it a Danaher Business Systems Leader because DBS is just Danaher's flavor of Lean Six Sigma, if you've ever heard of that, or lean manufacturing. And then rotation 2, after a year in Long Island, they shipped me down to Newport Richey, so north of Tampa, the aerospace device site. So I got to see more of how, devices were built in industrial applications. So it's kind of funny because the manufacturing environment up in Hauppauge with the life sciences is you got to gown up, put on a hairnet, put on shoe covers, beard cover, whole 9 yards before you go into the manufacturing area. Whereas in Newport Richey, you just kind of walk right out there and no one cares. People care. You want to stay as clean as possible because they are still kind of fine applications, but the filters are far more robust. The material we use in a lot of them, since they're subject to extraordinarily high temperatures and pressures on board aircraft, they use like this metal mesh in a lot of the filters, and so they're just much more robust. So the cleanliness standards are lower. I did 2 different cell lead roles there, so kind of a production supervisor supervising operators on the shop floor who are far older than I, have far more experience than I, so it's kind of intimidating. But cell lead of 2 different production departments, and then cell lead of the receiving department within the supply chain organization. So kind of got all the flavors of Paul Corporation as well as operations. And then I got a full-time role at Pensacola as a VSL, which is basically just an operations manager. I'm in charge of a few different manufacturing lines and product lines that we ship out to, that make media, which we ship out to our device sites. Common thread with all the products that are manufactured in my area, it's called Room 190, is that we do surface modification. So we take media that's manufactured from other areas of the plant. We put a special chemical on it, run it through a special process, whether it's going through an oven or a UV lamp or something like that, to initiate chemical reaction. After we coat it with a chemical, give it the properties that it needs, wash off all the unreacted chemical, and then we send it on its merry way. This is just a bunch of pictures of different sites. So top right is Hauppauge, bottom left is Newport Richey, bottom right is Pensacola. And then top left are just a couple things that have gone on in my career, a couple lessons learned. So that table of contents, it's just a long manual of how to receive different stuff. Since I was the cell lead of the receiving department in Newport Richey, it's very important just to document things in your career. So when you learn something and when you spend a long time knowing how to do something, make a procedure, make something so that it's kind of easier for people that come after you. I made this in mind because I knew I only had 6 months and someone else was going to take my place, and so it's like, all right, let's document this just to make it easier. Wrote a few engineering studies in the life sciences. It's very difficult to make a change, so literally I spent my entire year at Hauppauge working on one engineering study, trying to make a little change to the manufacturing process of some of our tangential flow filtration systems to make the operation a little easier on the operators when building it. But it involved changing how we pull vacuum from the urethane encasing, and R&D threw a fit because they were like, oh, it's going to change the effective surface area of the membrane, and you got to prove this, that, and the other. And so it just took a year, but we finally pushed through that change. Recalls, so calibration of flow meters is very important, because with filters, you always do an integrity test. We have specs that we need to meet in order to ship product to customer. And sometimes, the flow meter says, oh, this meets our spec, but then the calibration's due in 3 months and it turns out it was out of calibration. You don't know if that filter was good or not. So that happened one time at Hauppauge, where we had to recall a lot of product. And night shifts, manufacturing facilities run 24/7 in general, at least all the ones that I've been at have. So that's a fun part about working in manufacturing. Bandsaws, that's part of failure mode analysis on our tangential flow filtration systems in Hauppauge. So customer complains it's not working right, so we do a little test to it, and then we cut off the polyurethane encasing and then separate the layers of media one by one. In that particular TFF device there. There's 118 layers. So it's a lot of work to kind of see what's wrong with the device. And as you can maybe see, the guard on that bandsaw is up a little too high. So I actually cut my finger, had to have surgery to repair it. Thankfully didn't hit the bone, but yeah, workplace safety, take it seriously because, you know, you might cut yourself with a bandsaw and need surgery. So yeah, not a fun experience. So some lessons learned in my career so far, at least in manufacturing, maintenance workers or miracle workers. So if you ever go into manufacturing, get to know them, you know, be adaptable, do things correct the first time. Of course, you'll mess up, but if you know that there's a correct way to do something, even if it'll take more time, just do it. Because, you know, if you don't have time to do it right, then you have time to do it over. Live at Gemba, so that's a Lean Six Sigma DBS sort of thing, just meaning to go to the manufacturing floor, because a lot of the times you have an office or something or a cubicle where you can spend a lot of time behind your computer, but you know, that's not really where you want to spend your time. You want to go out to the floor, bring your laptop out there, bring your charger, just don't leave, because that's where stuff's actually happening. And then work-life balance, you know, I know a lot of y'all fresh out of college, going into your career, you know, work your ass off and you really try hard to make it in the company, but, you know, still take time to, you know, do the things you love, see the people you love, because if you don't, yeah, you might regret it. But yeah, very important. So I know I'm, you know, kind of, well, since we have the intermission after my presentation, I guess I can take a little bit of liberty, but I won't. So Danaher business system, about halfway through the slides here. This is the Danaher logo. So it's basically a C and a D intertwined, the C standing for customers, and then the D meaning Danaher. And then, you know, it's got our core value drivers in the middle there: quality, delivery, cost, innovation, in that order. The order is important. You know, people, plan, process, performance. So this is kind of the secret sauce that Danaher uses in their operations in order to kind of make the business continuously improve. And, you know, and then we have our, you know, our, so core value drivers are in the middle, and then we have our, I forget exactly what they're called, but there's sayings, those 5, you know, Danaher Kaizen is our way of life, innovation defines our future, we compete for shareholders. So, you know, very explicit about that. You know, it's like, you know, we aren't bashful about wanting to compete for shareholders. We want, you know, more people to invest in our company, because that's what Danaher does, is that it buys different companies. You know, Paul Corporation was acquired in 2014, and then we acquired the GE portion, the biopharm portion of GE, rebranded it Cytiva back in, I think, 2019. And, you know, we just acquire companies, make them better through integrating DBS into their operations, finding little changes that you can make in manufacturing processes to save a lot of money. So for example, one of the products on the product line on, you know, one of the manufacturing lines that we produce here in Pensacola, you know, it's worth $5 a square foot. We produce, you know, 7,000 square feet a day. Well, we take 7,000 square feet of raw material, and yields were at like 77%. So if we make like a 10% yield improvement, given that it's $5 a square foot, and you just do a bunch of calculations, but basically it comes out to a yield improvement that we made will save the company $500,000 annually. And we didn't really even have to invest that much time or effort. We just took a deep dive into different parts of the process and said, hey, why don't we process it this way instead of that way to prevent wrinkling of the membrane there, that therefore we don't have to, you know, scrap as much material, and we increase our yields and we save a ton of money. And we do that utilizing different Danaher business systems tools, which are just, you know, again, you know, I think I said it before, but it's just Danaher's way of doing Lean Six Sigma. And in the end, nothing about operations is overly complicated. Danaher CEO back in, I think it was like 2005, said all DBS is common sense vigorously applied. So that's really all it is. And, you know, by integrating that culture and that mindset, getting buy-in with everybody from the manufacturing engineers and the plant manager and the value stream leaders to the operators on the shop floor, you know, you create that environment of continuous improvement in manufacturing. You increase yields, you reduce defects, and you find ways to produce stuff better and faster to continue to deliver to customers. And that's evidenced by one of the factors is Danaher's stock performance over the S&P 500. To kind of put that over there, just if you continuously improve, you're going to continue to deliver that value to your shareholders. So now I'll get into a little bit of the fun part, some more of the technical stuff, even though that's not really my foray. So I kind of consulted some of the R&D folks on some of these applications. But again, a lot of this stuff is on the Danaher website. So I said I'd get to the beer part later. I know it's not necessarily life sciences or biomaterials, but it's still very interesting. So we kind of make these vessels not at the plant that I'm at, but it's sterile filtration for beer processing. So a lot of breweries are starting to invest in this technology since it's becoming scalable and economical, wherein we separate bacteria and yeast rather than just inactivating them through pasteurization. So, and in certain focus groups, the beer has tasted better, it has a better color, it's proven to have a longer shelf life. And as we evolve the technology around the filtration systems to have simpler cleaning methods, it gets a longer service life and reduce maintenance costs and make it more feasible for companies to invest in this technology for processing rather than just pasteurizing their beer. So actually getting on to more of the biomaterials related stuff in the medical field. So one of the RC Versupor line of products is produced on one of the product lines that I oversee at Pensacola, and that's used a lot in air and gas filtration. So basically, in the simplest sense, it just blocks liquids while allowing airflow. So a very important part of this media is maintaining viral and bacterial retention characteristics under extreme conditions, such as in autoclaving applications. You have to balance those retention properties, however, with flow rate, because it's like you want to block stuff, but you don't want to block everything. And you still need that process to move forward. So it's always a balance. It kind of reminds me of when I was working at a project with in interventional radiology my junior year at Hopkins. It was and they might still be working on this project, I don't know, but it's a medical device that kind of swells inside a blood vessel before an aneurysm to occlude blood flow before causing the aneurysm to hemorrhage. And you had to balance the properties of the device, the swelling characteristics, with mechanical strength, because you wanted it to swell to occlude the blood vessel, but you also needed it to be strong so that it doesn't fall apart and actually occludes the blood flow. Similarly, with filter media, you want it to block what you need it to block, but at the same time, you don't want it to block everything, because if it blocks too much, then you don't get a high flow rate and your process takes forever. So it's, that, and of course, I don't know as much the technical details behind the filter media as I did with the medical device project with interventional radiology. But anyways, that's a common thread in, as I'm sure all of y'all know, is, balancing different characteristics, different material properties to get what you want. And then, of course, as I mentioned earlier, also a lot of inline filtration for intravenous drug delivery applications. So an IV, IV's particulate removal. So things like even little glass fragments or pieces of plastic have been found to kind of travel in that IV stream. So our filter media can, retain that, because if you're injecting that into someone's, body, that's obviously not good. Bacterial retention, biocompatibility is another very important product and characteristics of all of our filter membranes that we use in life sciences, obviously. Low extractables. So extractables is a word that, I first heard when I started working for Paul, and I've heard it a ton since then. And it's basically just as a fluid flows through your filter media, whether it's gas or liquid or solid, well, hopefully solids, well, the solids that you don't want don't go through it. But, is anything going to leach off of the filter media itself? If your filter media has a high extractables or leachables profile, it destroys the point of it being a filter, because it's supposed to stop things from going through. And if things are, cleaving off of the filter media itself, it's like, what are you doing? So, of course, we have a lot of tests that ensure low extractables from our filter media, and then also used in a lot of different pharmaceutical compounding applications. And then most importantly, to highlight recent events, Paul has been part of a UK consortium, because we have several plants and UND or R&D facilities in the UK. So we've worked a lot on development and now distribution of the Oxford AstraZeneca vaccine, provide a lot of the bioreactors used in the in the scale up and production of that COVID vaccine, tubing, single-use bags. Some of the membrane that's produced, our Mustang Q membrane that is produced on one of the manufacturing lines that I oversee at Pensacola, goes into an ion exchange membrane device. So it captures negatively charged particles. Specifically, I think one of the adenoviruses that they capture for the COVID vaccine in order to kind of make the thing. I don't know the explicit details of it, but it's really cool that a lot of Paul's products have been used in COVID vaccine development, particularly over in the UK. So, that picture on the bottom left is one of our bioreactors being used. And then the one on the right is Boris Johnson visiting one of the Paul facilities. All those boxes in the background are the Paul logo. So, it kind of makes us proud to, be part of that process. And with that, I will end and thank everyone again for taking the time to hear the presentation, and thank you for inviting me. Of course, I'm happy to take any questions.
I wanted to ask a quick question about how did your material science training at Hopkins prepare you for a job like this?
So it's mostly just working together and problem solving. So the material science degree, the technical aspects of it, learning about biomaterial properties, mechanical properties of materials, electrical properties, solid state physics, all that stuff. I haven't really used much of that, unfortunately. However, when confronted with some of that information, it's not overly intimidating to me. So I'm able to kind of converse with the R&D folks and use that. But yeah, the large majority of the skills I got at Hopkins, which helped me in my career now, are problem solving and working together. So I know at least in the material science department, they have a leadership and management in material science and engineering class. Multidisciplinary technical teams was another class that I took, both with Professor Alyssa Eisenberg, who I believe still teaches there. She's a wonderful professor, and those types of classes were the things that really prepared me the most for what I'm doing now, especially leading people, because I have 12 direct reports across 24 hours, 24/7 shift schedule. So having to interact and deal with conflicting personalities, so a lot of those more soft skill classes, phonathon, learning how to communicate with people, believe it or not, that helped me out a lot in my current role. The material science degree itself, it's problem solving and kind of digging to root cause, seeing what's wrong and then collecting the data to support a hypothesis, because that's a lot of what you do in manufacturing is like, we're getting this type of defect and we don't entirely know why. So let's go investigate and drive to root cause, collect the data we have, analyze the data, maybe find the data we don't have and find a way to collect it so that we can analyze it and see what's going on here. So yeah, I hope that answers your question.
Yeah, thank you.
Any other final questions for AJ just before we break for our intermission? All right, if there's no more questions, if we could just get one final virtual round of applause as well. Thank you so much, AJ, for coming.
Yeah, thank you for inviting me. And if anybody has any further questions or you want to connect on LinkedIn, anything like that, I'm available, always free, always happy to talk. If you want a job, Hull Corporation, especially at Pensacola, we're hiring a lot, and we're actually building a new plant in near Greenville, South Carolina. That's going to be operational here in a few months. 400 some odd jobs down there. So I don't know how enthusiastic any of y'all may be about relocating there, but it's going to be a life sciences plant. So, and yeah, if you're looking for a job, we are hiring.